Good evening, dear students. I think you all can see and you can hear me, ne? So am I audible? Can you see my screen or the camera? Okay, very good. Thank you. Thank you. Okay, right. Okay, give me one second. Right. Okay. The session is ongoing on the YouTube as well. So if you have any signal problem, you can access into the YouTube. So search on YouTube, Science with Dr. Sir channel. Then you can access to the YouTube video as well. So if you have any uh, signal issues, you can rewind the sessions on the YouTube. Okay, right. So welcome you all to the seminar. So which is we are going to conduct in grade 10 lesson inheritance. Okay, so I am Dr. Sir. So I think you all know about me. So no need introductions about myself. So don't waste our time. So we have limited time to complete all the lessons. So I will directly go into the today session. Okay. So first, I think all of you have the tute with you. So please raise your hands. How many of you have tute with you? Because I have to make sure, confirm. Right. Okay. Okay. Most of you have the tute with you. So first I shared my screen. Then we discuss the important things in the tute, and then I will come to the whiteboard for the discussion. Okay. All right. So we want the session with the digital screen. Okay. I will share my iPad screen. Then we will discuss on the iPad. Right. Okay. Give me one second. Right. Okay. I think you all can see my screen now. So this is our tute. So free seminar. So topic is the inheritance. So grade 10, 20th unit. Okay. Right. So first they are given some introductions about our topic. So first you should see inheritance. So what they are telling, diversity among the organisms. So I will discuss something with you about the diversity regarding the among the organisms. Okay, so the biosphere is the home to vast number of the plant, animal species. Okay, so the biosphere, our surrounding biosphere, is the home for the vast number of plant and the animal. Okay, then species can distinguish by their external features which are inherited from generation to generation. Okay, so first clearly read. Then inherent characteristic are the features passed from parent to offspring, while individual of species share common traits. Okay, common traits, they still exhibit variations. Okay, right. So we will discuss first what is this concepts regarding this diversity. Okay, so they are telling this surrounding, our surrounding is the home for all plant and animals. Yes, we have, what are the animals? We have elephants, neither. So we have dogs, we have cats, neither. We have so much of plants, neither. So all of them are present in our biosphere. Okay, so plant, we know about the mango plant, neither. So scientific name as well you can use, neither. So mango, coconut, neither. Okay, so these are the some plants and animals. But do you think these all animals, though all plants have same characteristics? No, because elephant have different characteristic features, dog have different characteristic features, cats have different characteristic features. Okay, and mango tree, coconut tree, they are varies. What is that reason? Because they have individually different characteristic from their generation. Okay, they are getting different features, different characteristics from their generation. How they getting these features? How they getting? How they getting? So elephants, they have unique shape, they have unique some features. So they are getting that features from their elephant generation. Neither. So these these things, these external features or internal features. So most of the features they are getting by their generation. So their parents will give these characteristic features to their children. And their parents will be give these characteristic features to their children. Okay, all right. So this is about our things. Okay, so we have to write down some examples here. Okay, right. So we will write down here. So what is the examples we have to write down? First, you have a space here given here, neither. So in this space, please write down first. Okay, give me 1 second. Okay, so inheritance due, you have a space here, right, for the example, right? So give me 1 second to share my screen. So I have to share again and again. And so this is a little bit new for me also because this place is new for me and this system is new for me. So a little bit confusions will be happening either. So with the times. Okay, right. Okay. So in the examples, okay. So in the examples, you can write down now. You can see my screen either. So okay. So in the examples, please write down here. So now we are going to write down about the features of humans. Okay, human have write down. I'm also write down here. So you can see here, humans have diverse diverse body features. Body features. What are they? Body features such as what are the skin color, skin color. Then what are the features? Hair type, facial structures, and etc. Okay, so these are the some features in our human body. They are showing some uh, specific features for their humans. Okay, so skin color, it is unique for humans. Neither hair type, some unique hair types for the humans. Neither. So facial structures. So these are some diverse features, diversities. These are the causes for some uh, changes among the human as well, right? Okay, so other examples include the diversity in cat, parrot, neither roses, orchid, tomato, and brinjals. These are the some other animal categories and other plant category. They also have some diversities. Okay, right. Now we move on to the inherent characteristic, most common inherited characteristic in our humans. Now we are not going to discuss about the plant. Now we are not going to discuss about the animals. Now only we are consider about the human. So you can observe these pictures. Okay, first we are going to observe the pictures one by one. Okay, so in the first picture you can see there are so many people in a single frame. So in these people, please clearly observe about their skin color, skin tone. Okay, so some of them are very whitish, some of them are very blackish, neither. So their skin color tone is vary. Okay, so skin color tone is vary. Okay, so we can observe different skin color. So example, first we can write down here the skin color. Okay, right. So first examples we can write down skin color. Right. So I'm also going to write with you. So first examples, write down skin color. Okay. So what are the colors you can observe in this picture? You can observe white color, neither fair or dark. Okay, so example, you can write white people's, neither fair people or dark. Okay, so these are the some skin colors we can observe here. Okay, please write down with me. I'm not going to give a separate time to write down. Okay, so second example, second inheritance characteristic. So these peoples get whitish color or the black or dark color or the fair color. That is due to their parental inheritance. They are getting the parents characteristic. So if their parents are white, sometimes their children will be white. So that is from your generation. Okay, right. Then second about the ear. So you can observe clearly here, this earlobe is separated. So here there is a separation. Okay, but in this ear, it is not separated, it is attached. So this is the earlobe, type of the earlobe. It is important. Next, common characteristic features. Now see your ears. So my ear is, I think you cannot see neither. My ear is the attached one, not separated. So in my family, my mother and my father, their family generations give me this characteristic from their generations. So their unique feature, this is the unique feature for our family. So our family, all members have this type of ears, most commonly this type of ears. Okay, but observe your ears. So your mother or father have your same type of ears. So this is coming from your generation. Okay, right. But sometimes your friend and your earlobes can be changed. Why? Your friend and you are from two families, two generations. Okay, that is the reason, right? So now write down here, ear lobe type. Okay, so the second option is earlobe type, right? Okay, so here earlobe type, what are the types we have? We have, they are fused or free earlobes. Okay, fused or free earlobes, right? Free or fused earlobes. Okay, right. Second, third. Okay, third, we come to the hair. Okay, so look at here. This hair, they are colored. Don't think about the color. Please consider about the nature. So their hair, some their hairs are curly, some their hair are straight. So type of the hair also, the features coming from your parents. Okay, so hair hair type. Okay, third one, hair type. So it is, can be curly or straight. Okay, curly or straight, right? Curly or straight, right? So you can see here, right? Now curly or straight, right? Hair type. Then we are going to next picture. So we correct, we talk about the skin color, earlobe, and hair type. What is the other one? So here, next one, when you smile, you can observe some people, they have a dimple. Dimple means some they have a dimp. Okay, over their face. මේ පොඩි වලක් වගේ එකක් නේද? So they have in their face. So that is dimple. Okay, so when their face, they have a cheek නේද? Dimple or normal cheek. So this is the cheek. So here, this side, they have a dimple, they have no dimple. So this is the one common inherited characteristic. Okay, so cheek type. Okay, so next one is the cheek type. So what are the two types? We have dimpled or dimpled or normal. Okay, these are the two types. So what are the next one? So we will go a little bit faster. We have to complete all these two with today. So the nature of the hand. Okay, thumb positions. When you close your hands together, your thumbs, sometimes your right thumb will on the top or your left thumb on the top. So these are two natures. So please do that. Do that and look at what finger is prominent in your hand. So some of you have right thumb is prominent. So who has right thumb prominent? Please raise your hand. Who has this right thumb prominent? Yes. Who has left thumb left prominent? Yes. Yeah, you can see. All of you have not right side. All of you are not left side. So some of you are right side thumb prominent. Some of you have left side thumb on the top. Neither. So this is due to inheritance. This is due to generation to generation varieties of the features. So this is the next features we are going to write down here. So thumb position when finger are crossed. So fifth one, thumb position when fingers are crossed. Okay, so we have two types. So here you can write down the two types. Right thumb top or left thumb top. Okay, so these are the two types. So what are the next one? So we have to go about the tongue. So see these two tongues. Some their tongues they can curve. Okay, they can curve their tongue. They can fold. They can fold their tongue inside the mouth. Neither. So they have a ability to folding ability. Okay, so next sixth one we are going to write about the tongue folding ability. Tongue folding ability. Okay, so see that ability to fold or not. Okay, so tongue folding ability, uh that is two type so have or don't have, so no need to write down here. Next one, next ability, what is the next features? Straight thumb or curved thumb. So when you are show like this, fit කියලා පෙන්වන්නේ, so fit මචං නේද? So when you are do like this, your thumb sometime it go into straight positions. So some people have curved thumb නේද? So that is coming from your parental generation. So their thumb shape. Okay, thumb shapes. So what is their shapes? We have straight or curved thumb. Okay, straight or curved. Okay, so finally last features. So last features we tell about the widow's peak. So widow's peak means this is a triangular shape top position in your hair, facial top hair. So in your frontal area, you have a hair shape. So some of them have this curve shape hair, the widow's peak. Okay, we call as a widow's. Okay, right. So next one is the widow's peak. So you can see here widow's peak. Is it present or absent? Either we can write present or absent widow's peak. So tongue folding ability also we can write present or absent. Okay, right. So now I finish first chapter or first page. We are finished. Okay, so common inheritance characteristic features we discussed. Right. So now now you can see what are the common inheritance characteristic features among humans. Neither. Right. So these 8 are given in your textbook. So more than that, some features are available, but no need to refer because in your level, this is enough for you. Okay. Right. So next, we are going to discuss about the activity. I given an activity. So take a 2 minutes time and think about your family members. Okay, so there is a table to fill yourself. So think about few people, close relatives in your father's family and close relatives in your mother's family. So So it is better to, if you are take mothers, your mother have some sisters and brothers, no, take them as relatives. So your father also have sisters and brothers, ne? So take them for this filling of the table. So think about their skin colors, whether they have widow's peak or not, cheek type and earlobe type, thumb shape. So you all, you must, sometime you all may not all features you don't know නේද, but you remember some features නේ, ඔක්කොම දන්නේ නැති වුණාට පොඩ්ඩක් දන්න features ටික fill කරන්න. So you can fill these features with your past memories නේද. So you may meet them නේද several times නේ. So think about your family members and fill this table. I will give 2 minutes for you. Okay, so this is time for you, fill this table. Okay, think about your family members, neither your father's family relatives, your mother's family relatives, neither. Think about them and fill this chart, mean this table. I will give few minutes. 2 minutes enough, neither. Think about their skin color, put skin colors, and think about your widow's peak, think about their cheek type, think about their ear lobes, neither. Think about their thumb shape. You can fill this table. Right. How many of you finished? Please raise your hand if you finished. Okay. So give me 1 second. I will share the may. Okay. Okay. Take another 1 minute. So I will put the YouTube link into your WhatsApp communities. If you have a friends, so if you uh, want to join, you can share the links to them. So it will be helpful for them as well නේද. So තමන් තනියම ඉගෙන ගන්නවට වඩා දෙයක් නේද, යාළුවෝ එක්ක බෙදා හදාගෙන ඉගෙන ගන්නකොට ඒක yaadිනවා. ඒක ගොඩ යනවා නේද. So that is what I believe. So when I was at the A level time, so I share in my O level time as well, I share all the things with my friends නේද. When I have some secret notes as well, I will share for them. So I'm not think about myself. I'm not think about my results. I'm just share. I I always think about my friends. So යාළුවොත් ගොඩ ගියා, මමත් ගොඩ ගියා. So that is why I am now in these positions as a doctor, නේද? As a teacher. So I am doing two professions. So two valuable professions, නේද? So one is I am saving the lives of the patients. One is I am teaching my students and make them to neither make their future brights neither we can so these two professions are very valuable professions in our countries neither. So I have that opportunity to do both two works. Okay, right. So kaiwarga huati. So may now activity 20.1. So I think you all finished that. So skin color, you can see now, observe your chart. Okay, so think about your if your father are some fair dark skin, most of your father's family members also having that same skin tone. Okay, if your mother is fair, your mother's family members are going to fair most of the time. All of them have a same skin tone. Then widows speak, if someone have most of the time, other people's also having. Okay, right. So cheek type, if they have dimple, neither some other members also having dimple. So your chart is now giving some information for you. So what are the observation you can take from this chart? Most characteristic are passed from parent to offspring. Why? Your father's parents. Okay, that means your father's parents were තාත්තගේ amma or තාත්තා නේද? So their parents are common. So common for that family. So their features come into that all the children. හැම ළමයෙකුටම වගේ යාළගේ features ඇවිල්ලා තියෙනවා. So some amount of the features going to all the children. So most characteristic are passed from parents to offspring. Okay. So then second one is the new traits may appear. The new traits. New traits mean new features. New features can be happen. Okay. Sometimes if your mothers or fathers have some curly hair, but some people, sometimes your mother's sisters or mother's brother, they have straight hair. Sometimes it is differ. So new traits may appear which could be inherited from earlier generation. That means if your mother have a curly hair, but other members having in your mother's family, other members having what hair? Straight hair. Okay. So අම්මට තියෙනවා curly hair එකක් අනිත් හැමෝටම තියෙනවා straight hair එකක්. Why that? Sometime in your mother's parents family. So අම්මගේ තාත්තගේ පවුලේ හෝ අම්මගේ අම්මගේ පවුලේ. So someone has that curly hair. So it is come from the older generation. නේද? So older generations features also can appear in this new generations. Okay, so that is the observation you can take from this chart. Okay, now conclusion is inherited traits can sometimes skip generation but remain present in the genetic makeup of the family. Sometimes skip වෙන්න පුළුවන්. So if some feature it can be skipped from one generation, but sometimes it can be appear in next generation. Sometimes ඔයාලගේ අම්මලට නැති feature එකක් ඔයාට තියෙන්න පුළුවන්. එහෙම නැත්නම් ඔයාට නැති feature එකක් ඔයාලගේ ළමයින්ට තියෙන්න පුළුවන්. So that can be skipped නේද? But their genetic makeup is present in your body. That is the one. නේද? That is the reason. Your genetic information is present inside always your body. Okay, but it is not appeared in yourself, neither in your body. Sometimes it will be present in your brother's body, neither. Sometimes you have hair type කියලා හිතන්නකෝ. So curly hair වෙන්න ඕන genetic makeup එක your body you have, but it is not prominent in your body. You have straight hair, but your brothers have curly hair. So sometimes your children can get the curly hair. Why? Your genetic makeup is inside in your body. Okay, right. We'll discuss those things later. So up to now, are you clear all the things? So if you got all the things, please raise your hands. Right. Okay. Well done. Well done. Right. So this is about the first introductions about this lesson. Now we are going to see some about rare inheritance characteristics. So I have to share my screen again. So give me 1 second. Right. Okay. So now we move on to the next part. Okay. So rare inherited characteristic. Okay. So see in your tudes. So some say rare things. It means මේක හැමදාම දකින්න හම්බෙන්නේ නැහැ. Very rare. ගොඩක් කලාතුරකින් some of your friends have these features. නේද? Right. What is the first features is the syndactyly. Syndactyly means fused fingers or toys. That means your friends have fused fingers. These two fingers are fused together, or these two toys are fused. So this nature is coming from their parents' generation, but all of their members are not having this feature. It is skipped from one generation to generation. Some of offsprings will show this feature. So that is rare characteristic feature. So inheritance feature syndactyly. Okay, syndactyly. Okay, first finger. Then second one is the polydactyly. Polydactyly means they have more than 5 fingers in one hand or one toe, leg, neither. So extra fingers or toys. So you can see 1, 2, 3, 4, 5, 6. So usually people have 5 fingers in hand, but some people they have 6 fingers. So how many of you show this polydactyly friends in your school? Please raise your hand if you see some polydactyly friends in your schools. Yes, yeah, very good, very good, very good. Because most of you have seen these features in your school level neither. Because a rare feature, but 1 or 2 people can present in every schools. Okay, right. So next one is the albinism. Albinism means lack of pigmentation in skin, hair, and the eyes. So they have lack their pigmentation in the skin, hair, and eyes. So they all are appear white. All the skins are white. All the hair are white. Eyes are white. Neither. So they are look like me Sudha people. Sudha people. Okay. All of them are white, but Sudha people are not albinism people. Their eyes, their hairs are blackish, but their skin tone is somehow white. But that is not due to albinism. But when they albinism people, when you see all of the skin, all over the skin, all over the hairs are white. Okay, so how many of you see these type of friends in your schools or in your areas? Yes, most of you see neither. Okay, well done. So this is albinism. So this is a one rare inherited feature, right? Next one is the eye color. Okay, eye color means some of them have a blue color eyes, some of them have a brown color eye. Okay, blue color and brown color eye. Now, I mean, in my school also have one friend. May she has she has blue color eye. So අපි ආදරයට කියන්නේ මේ බලල්ලස් තියෙන යාළුවා කියලා නේද? So බලල්ලස් mean the පූසා නේද? The පූසා has some blue color eyes වගේනේ නේද? So ඒ වගේ eyes features they have නේද? So how many of your friends or how many of you have this මේ blue eyes? Yes. Yeah, few of you have blue eyes. So that is not a disease. Don't worry, that is not a disease. That is a special feature. It is coming from your generation. So it is very beautiful sometimes. So it is coming from their parent generations. Okay, right. Now we'll move on to the next part. Right. So we have transmissions of inheritance characteristic. So how these inherited characteristic are transmissions? Now, inheritance is common to all living organisms. Okay. Then plant, animal also inherited traits like skin patterns, beak shape, fruit taste, neither flower color and plant height. It will be transmit from generation to generation. Now you have an assignment. So select few plants and or animal in your garden. Okay, select some plants and animals in your garden. Then record their characteristic have remain unchanged over the time. That means ඒක මේ බල්ලෙක් හරි පූසෙක් හරි ගන්න නේද? So බල්ලෙක් හරි පූසෙක් හරි අරගෙන මේ ඒ බල්ලගේ පූසගේ මේ ෆැමිලි එක දිහා බලන්න. බල්ලගේ අම්මා කවුද? බල්ලගේ තාත්තා කවුද නේද? So ඔක්කොම හොයන්න. So when you look at that, you can observe some features that is not change from their parents to මේ their daughters or sons නේද? Their offsprings are carry that feature from their parents generation neither, so that is unchanged. Sometime if you are take a fruit plant generation, so if you have a one jackfruit tree in your garden, so see about the other jackfruit coming from that tree neither. So sometime your කොස් ගහක් තියෙනවනේ, ඒ කොස් ගහේ පැලයක් තව හදනවා. So see about what are the features unchanged. සමහරක් විට the taste රසය අර ගහේ තියෙන රස මේ ගහෙත් තියෙනවා. So that is a neither some features unchanged. Neither. So you can observe these things in your garden level. Okay, right. Now we move on to the examples of inherited characteristic in plant and animal. Okay, so examples of inherited characteristic in the plant. What are the plants examples you can see? What are the plants example? Flower colors, the color of the flowers, fruit taste. So these are the and seed shape. Some of them have round shaped seeds. Some of them have sausages like shape. So the shape of the seeds, color of the flowers. Then uh, fruit taste, they are vary. Okay, right. Write down with me. So plants, their flower colors, then fruit taste, then seed shape, seed shape, then what are the other things? Plant height. Some plant generation, they are very tall. Neither. Some plant generation, they are very short. So height. Neither. Plant height. So today my handwriting is not very good. මට හිතගෙන මෙහෙම ලියන්න බෑ වෙනද මම වාඩි වෙලා ලියන්නේ so when I sit in the good position I can write clearly නේද but now I am standing so I cannot write good handwritings. Okay, so don't worry about that. So what are the features you know about the animals? So animal features, what they have, inheritance features. So we already discussed about the human features. So that features you can write down into the animal. So skin color, then ear shape. Yes. What are the things we discuss about the cheek? Cheek is not common in the all animals. So what about other features? Yes. Tell me. Wing structure. So wing, if they are present wings, what are the wing structures? They are common for their generation. Wing structures, what are the things? Neither. The color of their and their body, ear shapes, then wing structures and shape of the eyes. Eyes, neither. Type of eyes, type of eyes. So these are the some characteristic features, inherited features among the plant and animal. Okay, so these are the example. Right. Now we move on to the non-inherited characteristics. Okay, non-inherited characteristic feature. Right. So non-inherited characteristic feature mean some features are not going to transmit from one generation to next generations. So one of my friends may ask, so may the cricket players we have some cricket players so who are the cricket players we know? The good bowlers. So Lasit Malinga, take the example for Lasit Malinga. Lasit Malinga is a good bowler so in the Lasit Malinga he are using, if he using right hand, so his right hand little bit longer. Why? He is usually using this hand. So his left hand is shorter little bit than right hand. Right hand is little bit longer. So that feature can be transmitted to the daughter or son of the Lasit Malinga. No, that feature cannot transmit. That feature happen due to his current activities. So it is only present, that feature is only present in the Lasit Malinga, that is not going to transmit in next generation. So if you think about some bodybuilders, okay, some bodybuilders, they have a strong muscles bulk. So so strong muscles bulk. So strong muscles bulks they have. So uh, are their children are their children having this type of muscles bulks? Yeah, their children are usually sometimes the father is the very fat bodybuilder. Lamai very thin. නේද කෝටු කෑල්ලක් වගේ ළමයි නේද? So that is not going to transmit from one generation. Then other feature, so your mother has a scar in the abdomen. Okay, so your mother scar mean කැල් surgery එකක් කරපු පාරක් තියෙනවා නේද? කපපු පාර තියෙනවා බඩේ. Okay, so your son or their sons, okay, ඔයාලා එහෙම නැත්නම් ඔයාලා නංගිලා මල්ලිලාගේ that මේ feature එක තියනවාද? scar එක තියනවාද? That is not going to appear in next generation. So that type of features we are called non-inheritance characteristic. Okay, right. So we'll write down with me. So what are the non-inheritance characteristic features? Okay, right. So I will share the screen again. Right. Okay, so non-inheritance characteristic features. Okay, the traits that are arise due to environmental factors, nutrition or activity are not inheritors. That means අර මං කිව්වේ ලසිත් මාලිංගගේ day to day activities නිසා ඒ hand එකේ වන change එක that is not going to transmit for next generation. නේද? So sometime nutritionally some people they are having get more nutrition, they will fat, but sometimes their children are very thin. නේද? So that is due to their environmental changes. නේද? So these features are not going to transmit to generation. Okay, right. So examples you can write down here. What are the examples? First example, growth of muscles. So growth of muscles due to exercise. Okay, due to exercise. Right. So when they exercise and when they having more muscles bulks, that is not going to appear in their children. Okay, so then another one is the corn toys. Okay, corns on toys. In the toes, some people they have corn mean podi අර මේ ගැටිති වගේ තියෙනවා. Okay, so that is happen due to the environmental factors. ඇඟිලි වල කකුල් වල ඇඟිලි වල ගැටිති වගේ තියෙනවා මේ corns on their toes නේද? So that feature is not going to transmit to next generation. Then the scars in the skins. Okay, scar on the skin. Scar on the skin. On skin, right? So scars on skin not going to transmit to next generation. So you can see those things as a non-inherited characteristic features. Okay, right. Up to now, we talk about all the inherited and non-inherited features. Okay, right. Now we are going to talk about the Mendel, Gregor Mendel. Okay, where the cursor? Okay, sorry, right. Now, here Mendel, Mr. Mendel. So he is the father of the genetics. Okay, he is the father. But the sad news, Mendel innawani. The Mendel, initially, he told these things. What we are going to learn. අපි දැන් ඉගෙන ගන්න යන දේවල් ටික එයා කිව්වේ ඒ කාලේ. He told all the things. But people not believe those things. මිනිස්සු ඒක විශ්වාස කරේ නැහැ. So he is die with sad. Okay, so some books they are writing the story of the Mendel. So Mendelගේ සමහර දේවල් මිනිස්සු පිළිගත්තේ නැහැ. So they told Mendel මෙයා පිස්සෙක් මෙයා කියන දේ අහන්න හොඳ නැහැ නේද? So that type of blaming is came to the Mendel. But after the death of the Mendel, after prolonged time, they find out these things as new things. Okay, මේ මෙන්ඩල් කියපු දේවල් පස්සේ කාලේ කියලා අලුත් දේවල් විදියට හොයාගත්තා. But some people know already these things already given by Mendel නේද? Mendel මේ දේවල් කියලා තිබුණා කලින්ම. He told all the things නේද? So then later they discuss and they plan to give the credit to Mendel, who discovered all the things. Then they name this Mendel as a father of the genetics. Okay, so Mendel is the father of the genetics. Some books they are telling the Mendel is a Swami. So he was a Swami. Can පූජකවරයෙක් වගේලු ඒ කාලේ හිටියේ. So that's why people not believe these things at the uh, at his current living state, but after the death they find out, they investigate these all the things, then only they believe the Mendel's concepts. Okay, so who is the Mendel? Mendel is an Australian scientist. Okay, so first to study the transmissions of inheritance traits. Okay, he is the celebrated as father of genetic. Okay, now we should know about something key terms to remember. Okay, right, so this is the key term. First key term is inheritors characteristics. So we discuss these things, but we have to write down here. Then we come to the Mendel things later. Right. Okay. So first inheritor characteristics, write down about the inheritor characteristics. Now we know what are the inheritor characteristics. So write down here traits, that is the traits, okay? Traits passed from one generation to the next generation. So if the traits pass from one generation to the next generation, we call as a inherited characteristic. Okay, what is the non-inherited? Non-inherited means they are not going to traits, not influenced by the environmental factor and the lifestyle. They are not going to transmit from one generation to next generation. So write down here traits influence. Okay, influenced by environmental factors and lifestyle. Okay, so they are not going to transmit. Then we have a dominant traits. Dominant traits means features which are going to dominant in our body. So write down about this. Then later we will discuss briefly about this dominant traits. Okay, so only for the key term remembering purpose, write down this dominant traits. Okay, a traits expressed traits expressed in heterozygous individuals. So traits expressed in the heterozygous individuals. Okay, right. So heterozygous individuals, right. Then what about the recessive traits? Recessive traits mean, what are the recessive traits? You know, neither. So what are the recessive traits? These are the traits expressed only in the homozygous individual. Okay, expressed only in homozygous individuals. Okay, right. So you know about now heterozygous, the individuals, if they have expressed the traits, we call as a dominant traits. Then if the traits is only present in the only expressed in the homozygous, we call as a recessive traits. So don't stress out about these words. We will learn these words clearly in the next part of our discussion. Okay. Okay. Where is my screen? Okay. You can see my screen, neither. Right. Okay. Little bit confusions will happen there. Okay. Right. So, Mendel experiments. Now, we are going to see these things in our notes. Then I will come to the whiteboard and I will discuss with you. Okay. Right. Mendel's experiments. First, Mendel studied may, this, may, pea plants. Pea plants means pisums stavum. Sorry, setium. Okay. So pea plants, we usually in the term we are telling about the pea plants to understand this inheritance. Okay. So Mendel's do some experiment using these pea plants. Okay. So these pea plants, why he use මේ pea plants mean අර ගෙවතු මෑ means මෑ කරල් මෑ short මෑ plants in the මෑ mean මෑ කරල් කියලා අපි කියන්නේ සිංහලෙන් නේද? ගෙවතු මෑ කියලා කියන්නේ චූටි ගස් ගස් වගේ හැදෙන චූටි plants නේද? So මේ ඒවා තමයි pea plants. Okay. Why he use these pea plants? First purpose is it is easy to grow and eats quickly. So it is easy to grow and yields quickly. That means we can take the seeds from the plants quickly. Neither. And it is growing quickly. Then second one is the pure breeding plants with unchanged characteristic over the generation. That means they're pure breeding plants with unchanged characteristic over the generations. That means if they have some features in their pure generation, that will not change among that generation. Then observe contrasting traits. Example, yellow, green seeds, tall or short. We can observe the changes, variations among their generations. So we observable contrasting traits are available in that pea plants. Okay. Second one is the naturally self-pollinating, but cross-pollination also possible. Okay. Right. Second one is the offspring can be obtained to study inheritance pattern. So their offspring can take to study about their generations. Okay, right. Now I will move on to the whiteboard. Okay. So why these Mendel take these pea plants to study? First one, it is easy to grow. So in any weather condition, in any area, it can easily grow. Okay, so that is one purpose. Then it is we can take their seeds or their leaves quickly. Then pure breeding plants with unchanged characteristic over the generation. If they pure, it means if you if you take short plants in a pure area, they have some only shorts, they not mixed with tall. Okay, right. So we can take the pure breeding plants. Okay, then observable contrastings are available. That means if you want to check tall and short plants, so that features can observable in the two generations or the among the single generation, you can observe the changes. Okay, right. Then naturally self-pollinated, but we can do the cross-pollination with our own. Okay, then it is useful for these inheritance studies. So now Mendel proceed for study tall and short plants. Okay, so Mendel take a short and tall plant. I will now tell me about this practical Mendel did in his garden. Okay, so he did this in his garden. Okay, so he take a very tall pea plants. Okay, so this is, think about, this is the pea plants. Okay, this is tall one. Okay, so this is the short one. So this is short pea plants. Okay, so these are two generation. So he take this tall one and he take this short one. Okay, then he did some experiment. He take this as a parent. parent means මෙයාලා තමයි දෙමාපියෝ. So මෙයාලගේ දරුවෝ ටික we have to see in the next generation. So these are the parents. These are the parents. So parents mean they are the one නේද? May produce they are going to produce the seeds, and from their seeds they will go on a next generation. So now this is the parent. So Mendel put this generation as a P1. So this is the P1 generation. Okay, that is the parental one generation. Okay, right. Now they go into second cross pollination. Okay, so second option, they are going to cross pollinate. So they are deposit the tall plant pollen on the short plant pollens. Okay, so they have a flowers. So in these flowers here also it has a flowers. So here going to put the tall plant pollen on a short plant stigma. So tall plant stolen in a short plant stigma. So this is the cross pollination. So he did cross pollination, right? So what will happen? Now they are going to give the next generation. Okay, now they are going to give the next generation. So in next generation, what can be observed? Yes, what can be observed? Their next daughters and their next generations will be appear there. Okay, give me 1 second. My lap is going out of the charge. Okay, right. Parents are here. Now Mendel observed that all their next generation plants are not short one, they all are tall one. Why that? Mendel observed these things. Okay, so they are all next generation plants are tall plants, not the short. Okay, they all are tall. Why that? Mendel think about that. Okay, we also having that problem. So all their next generations are tall. Okay, these are tall. This is tall. This is short. But their next generation, by the pollination among the tall and the short, they produce only the only tall plants. Okay, they produce only tall plants. Okay, so that generation Mendel label as a F1 generation. This is the family one. Okay, so this is the F1 generation. Then next, he's going to F1 generation. All offsprings were tall. They all are heterozygous. Okay, they all are heterozygous. We will discuss those words later. Now I will going to tell about the fourth point. They self-pollinated among this F1 generation. That means they are going to take a two offspring from the F1 generation. So now they are taking the F1 generation, two plants. Okay, two tall plants. Okay, two tall plants. Now they are taking from the F1 generation. Okay, I think you can see my board neither. All right, very good. So this is tall. This is also tall. Now they are going to self-pollinate among these two. Okay, so then they will observe some features of next F2 generations will be present. So the F2 generations, they have, okay, they have four members in their next generations. So one is, there are three plants are very tall, and three plants are tall family. Okay, but one plant, this is tall, but one member, they were short nature. So these three tall and short one in the next generation. What the name we are telling to that generation? We are telling to that generation that is the F2 generation. So this is the F2 generation. Okay, so this is the experiment done by the Mendel. Okay, so Mendel did this experiment in his lab, okay, in his garden. So what he did, he take the pea plants, one tall plant and the short plant. He take homogeneous. Homogeneous means the same features appear in their same alleles. Okay, same alleles are present in there. Don't worry about this word alleles. I will explain later. Okay, right. So in the parent plants, they are taking short and tall. So tall is capital T, capital T. We express by the capital T, capital T. Short plant we express by the simple T, simple T. Okay. So among these P1 cross pollination, capital T to simple T, then they are all next generation. So they having 4 members. Okay, 4 members in the F1 generation. F1. Okay, so in this F1 generation, 4 members, all 4 are capital T, simple T, capital T, simple T, capital T, simple T, capital T, simple T. That means they are heterogeneous. So these are heterogeneous generation. Then cross-pollinated amount capital T simple T one plant with capital T simple T heterogeneous self-pollination. Here self-pollinations we did. Then in these self-pollinations, they will produce 3 whole plant and 1 short plant. So among these plant, we can observe 1 capital T, 1 capital T, capital T, simple T, and simple T, simple T. So these are the things observed in the Mendel's experiment. I know you all are confused about this capital T. What is this capital T? Mono බම්බුවක් දානවත් දන්නේ බෝඩ් එකේ අපිට නම් තේරෙන්නේ නැහැ. Okay, don't worry. I will explain step by step. So only this discussions purpose I put these words. So this is done Mendel in his garden. So we will learn about what is this capital T, what is about the simple T, simple C. So wait until the finish of until next part of our session. So we will discuss what is this capital T, what is this simple T, what is this F1 generation, what is this F2 generation, how they are produced, all the things we will discuss. So don't stress out. Okay. So this is the things happen in the Mendel's experiment. Okay. Right. Now, simply we can write down the observations. So we will go to write down the observation first. Then we will come to the again discussion for about these things. Okay. So go and write down the observation. So I will share my screens. Okay. Don't worry about these words. So I 100% sure you all get complete knowledge when this seminar is finished. Okay. Everyone, definitely you may get all these concepts crystal clearly. Okay, so don't stress out. So I will help, I will do my maximum for you. Okay, right. Then observations. What is the observation is dominant traits. Okay, dominant traits marks recessive traits in the F1 generations. Okay, that means dominant traits, that means tall, masked. Okay, mask mean they mask the recessive tall in F1 generation. That means the F1 generation they don't have a short one. කියන්නේ යාළ වහගත්තා. ඒ generation එකේ F1 එකේ short කට්ටිය නැහැ. Only the tall people. නේද? That one dominant traits, that means the tall nature, it is masked the recessive traits, that means short in F1. Okay, so in F1 generation, right? So that is the first observation. So dominant traits, that mean tall plants, they mask the recessive plants in the F1 generation. So second observation is recessive traits. Recessive mean the main short one. Okay, recessive traits that means short reappear in F1, F2 generation, that means the next generation, F2 generation. Okay, right. Then that means recessive is came back again in the F2. Yes, recessive is the short one. So short one came back again. So don't worry about, I will explain again. So only write down these things. So don't stress out. Right. Then last observation is each traits, each trace determined by two factors, two factors, two factors. So write down in the bracket two alleles. Okay, two alleles. Now the word is coming alleles. Alleles. Right. So this is these are the observation. Dominant traits mask the recessive traits in the F1 generation, and recessive traits the short reappear in the F2 generation. Each trace determined by two factors. Right. Okay. So that is enough. So now we will write down the what is the allele. Then I will again come to this discussion. Okay. Right. Okay. No, don't stress out. We will write down the what is the allele. Okay, right. So write down the what is the allele. Allele are, alleles are, write down with me. Alleles are different versions. Okay, different versions of the same gene. Okay, different version of the same gene. Okay, they determine variations variation in inherited traits. They determine variation in inherited traits. Okay, inherited traits. This next point, there are two type of alleles. Two type of alleles. Okay, one is homozygous, other one is the heterozygous. So complete the note about the what is the allele. Okay, so what is the allele? Alleles are the different version of the same gene. They determine the variation in the inherited traits. There are two type of alleles. Okay, so what are the two type? First one is homozygous, second one is heterozygous. Okay, so homozygous, write down about the homozygous, same allele. Homozygous means same alleles. So in the bracket, you can write down capital T, capital T. That means two alleles are same. Oh, simple T, simple T. Right. Then heterozygous mean different alleles. Different alleles. Examples, we can write down capital T, simple T. Okay. All right. I think now you all are in a confusion state. Don't worry. Okay. So complete this note and come to the discussion part. Okay. Right. I will go to the whiteboard now. Right. So now we will go to the discussions. Now you know about some words about the, we learn about the alleles, and we learn about the homozygous and heterozygous alleles. Okay. So allele mean they are the one version of the gene. Okay. Gene mean gene is present in our chromosomes. Chromosomes, we have a DNA. So in our DNA or RNA, both of them present in neither. So especially in the DNA, they have a special type of area. That area determine one feature. Okay, that means in our body we have so much of DNA. So in the DNA, in the chromosomes, so much of chromosome. In the chromosomes we have DNA. In the DNA, one specific part, they are determined our some features. Okay, some features they are determined. That feature, that area we call as allele. Okay, that area we call as a allele. In the next later part of our discussions, we will discuss these things. Okay, don't stress out. So that is we discuss in the next part. So that is we call as a allele. So if someone has a dominant feature allele, dominant feature means if you consider about the two features, one is the more prominent one, most prominent one, other one is a very less feature. Okay, so more prominent feature we call as a dominant feature, other less prominent feature we call as a recessive feature. Earlier also we write down in our note, we written in our note what is that about the dominant traits and the recessive traits. Now read about the dominant traits. Traits expressed in the heterozygous individuals. Heterozygous mean, now you know heterozygous mean, if you are consider about the capital T simple T, that is heterozygous. Okay, that is heterozygous. Why? These both of them are different. So this is heterozygous. In this heterozygous individual, what the feature they are telling? So I will tell this capital T is tall, simple T is short. So what their outcome? The capital T, simple T, what their heterozygous state, what their feature is they are going to express. What the features they are going to express. That express feature, what the features they are going to express when they are in the heterozygous state, we call as a dominant feature. Okay, that means they have a capital T allele also, they have a simple T allele also. Capital T allele tell about the tall nature, simple T allele told as a short nature. So in the heterozygous state, when they have a capital T and the simple T together, what feature they are going to express? That is the dominant feature among that two. Okay. So here they are going to tell about the tall. That's why I put capital T, simple T in this generation. Okay. Right. Now you know about the dominant. So if they are in the homozygous state only, they are prominent other recessive feature is expressed in only in the homozygous nature, that is short nature. Okay, right. So now you know these are the two types. So capital T is tall, simple T is short. Okay, capital T is short, tall, simple T is short. So these are the two features. So among the capital T is the dominant one. This is the dominant feature. This is the recessive feature. Now you can see capital T is the dominant. So when they have capital T parent, that is tall. Okay. So that is tall. That's why I put it is as a tall. So when they have a simple T, so in the homozygous state, so they are what? They are short. If they have only the capital simple T, so they are short. Now you can observe easily capital T, simple T, when they are the cross pollination, when they are cross pollination, their next generation, they have only tall natures. They have only the tall people. Neither. So they have only the tall P plants. Okay. So capital T simple T, capital T simple T, capital T simple T, capital T simple T. Now they are in the heterozygous state. In the heterozygous state, what are the features they are telling? Express. They are express the tall nature. So the dominant feature is they are going to express. That's why these all plants are now tall. Okay. Now these all plants are tall. Okay. Are you clear? So if you got the idea, please raise your hands if you got the idea. Right. Do you have any doubts? So if you have any doubts, please put into the chat box on your Zoom meeting. So if you are in the YouTube, put your uh, questions into the comment section. So I will reply them in the interval. Okay, right. Okay. So this is happening here. So then we move on to the F2 generation, F1 generation, people cross-pollination, self-pollination. So capital T, simple T cross-pollinations happen here, self-pollination. So self-pollination, they provide capital T, that is tall, capital T, simple T also tall, capital T, simple T also tall, but simple T, it is expressed short nature. So this is observe Mendel on his F2 generation. Right. Now we have to draw this all the things in your book, but I not given the spaces in your tube. Okay. So I forgot to put the space in your tube. So you have to write down this all the things in your book. Okay. So take a paper and write down these things in your book. Okay. Draw these all the generation. So first draw this side, then draw that detail in lower part. Okay. Right. So I will give only 5 minutes. Please take a sheet and draw these things. Then we can easily understand what will happen here. Okay. So I think you cannot may see here neither. So I will write those things in this area. Okay. Right. Capital T is telling tall. Simple T is telling short. Here the capital T is dominant feature going to express. Okay. Right. So draw these all the things in a sheet. I will give a 5 minute time. So draw and complete your note. Okay, I finish. If you finish, please raise your hands. Not yet. Okay, you can take another few minutes to complete this drawing. Yeah, So this is the experiment done by the Mendel. So we have to understand the inheritance concepts through this experiment. That's why I asked to draw. Okay, please draw clearly. Okay, so you will have an interval around 6:45. Then again session will be started around 7 p.m. So get ready your dinner or your cup of tea around 6:45. Okay, now the 6:30, within next 15 minutes you can have a, after next 15 minutes you can have an interval. So around 6:45 to 7, I will give the first interval. So then we will give the next interval around the 8. Okay, right. So no need two intervals neither. Around 9:30 we can finish. Okay, I will consider about the second interval. So you can take the first interval around 6:45. So keep in mind. Okay, so if you finish the drawing, please raise your hands. So seminar will be, yes, it will be happen around 9:30. So after 9:30, if the sessions finish or the lessons finish, I can end the sessions, or I will continue the teaching until finish uh, around 10. So maximum around 10 we can spend. So more than that I cannot do. Okay, right. Mm, I think we can finish all the things. Now we know about these things. Now go to the next part of our uh, discussion, tute. Right, so I will take the tute. So in our tute, uh, next part is telling about the monohybrid pros. So they telling these pros, these experiments again. Okay, these experiment again we have to discuss in a different way. Okay, so example, capital tall was short plants. Okay, so monohybrid cross mean mono single feature. Single feature mean tall or short. So depending on the height of the plant, only consider, and you are going to check that only one feature among the generation how is transmit. Okay, so like this. Okay, right. So I will explain here. Now, what is the Mendel did? Mendel did, he take the capital T tall plant, capital T, that means the homogeneous tall plant and homogeneous short plant. Okay, so these homogeneous generations, now he did cross pollination, and first generation, all of them are tall. All of them are tall. So where is the short? Where is the short nature? Short nature is skipped from this generation. Okay, short nature is skipped from this generation. Then it is again, we take the F1 generation, capital T, simple T, capital T, simple T, two tall plants. That means we take heterogeneous two plants and we self-pollinated among them. So again, we get capital T, simple T, capital T, capital T, capital T, simple T, capital T, simple T. Again, we got simple T, simple T. Okay, so this is what happened here. So the parental plants, we have the nature of the tall and short both, but in the first generations, we don't have the short plants, but in the next generations mean in the F2 generations, again, short plants is reappear. So this is the experiment done the Mendel to identify how these inheritance features transmit from one generation to next generation. So he identified there are alleles, that alleles are the one version of the genes. So they are the responsible for these features. So if they have the dominant allele, they will give the dominant feature. If they have a recessive alleles, they will give the recessive features. Okay, so that is the concepts we should clearly understood. Okay, right now I will going to erase all the things. Now we are going to monohybrid pollination. Monohybrid cross. Okay, monohybrid cross mean monohybrid pollinations among the generations. Okay, right. So examples they are telling in the tall was short plants. Okay, so example is tall was short. So in the you have a space given there name. Okay, in the space write down with me the space tall is capital T, capital T. Okay, short we taken simple T, simple T. Okay, so we take the capital T and simple T as a short, capital T as a tall. Okay, this is tall, this is shorts. So these all together, these are homozygous alleles. Okay, so these are the parents. These are the parents. These are parents. Okay, parents have homozygous alleles. Okay, so now in the first point, in the point one, P1 generation, they are tell about the P generation. Capital T cross with the simple T. That means homozygous dominant cross with homozygous recessive. So this is the one homozygous dominant. So if you want, you can write down here. This is homozygous dominant one. Capital T is the dominant family. Okay. So this is the one homozygous. So if you write down this type, that is better. This is recessive. Okay. So in the P generations, they are going to tell their P capital T, simple T is going to cross. Okay. Right. So capital T cross with simple T. Okay. Right. So capital T cross with simple T. Okay. Right. So write down here, there is space in the P generation. So P generation කියන තැනට යටින් there is a space. So write down there. Okay. Medically aganda. Here we are doing cross pollination. Here we are doing cross pollination. Here we are doing cross pollination among these two parental plants. Here we are doing cross pollination among the parental plants. Okay, right. So then what happened to them? We'll see. Okay, look at here. Okay, look at here. Now finish writing. Okay, so look at here. So capital T, simple T. Now capital T means they have two alleles. They have two alleles in their generation. So if I separate that two alleles, I can separate capital T one allele here and capital T another allele here. Okay. Now, among this other family, I will put double line here to separate this family from this parent from this parent. Now, in our next parent also having two alleles. So they both two alleles are simple T and simple T. Now what I am doing, I separate these alleles from the parents. Okay. So parental alleles I separated. Okay. I separated. So now think these two alleles can combine one of this parental, one of this maternal or paternal. Neither one of this maternal allele can cross with one of the paternal allele. Neither. So one parent allele can cross with or combine with one other parent's allele. So this will be happen here. So we'll write down here. So this one can go with this simple T. So I put here the when they combine together this capital T with this simple T, they can provide children or the next generation. It is capital T, simple T. Okay, right. So this parent, okay, this same allele can go with other allele of the next parent. So it also can produce capital T, simple T. So now we finish about this allele in this parent. Okay, right. He has another one allele. It is not yet crossed. So we have to cross that allele with next parental alleles. So this allele, now I'm going to cross with this allele. So it is crossed with capital T, simple T, they will provide. So this is cross with next allele as well. So this is going to provide. So capital T, simple T. So this is the one F1 generation. So this is F1 generation. How about their features? How about their features? Are they all tall? Are they all short or short and tall separately? Yes, you can see here. This is tall. This is tall. This is tall. Why? The dominant feature is capital T. Ne? So if they have dominant one allele, so they will definitely express that feature. So all of them are tall. Neither. Right. So what they are telling in our next part, F1, they are telling in the number 2, F1 generation, all the heterozygous tall phenotypes. Okay. There is a space given here. So write down there. Write down all the F1 generation, all the F1 generation, all the F1 generation having capital T, simple T. So capital T, simple T, capital T, simple T, capital T, simple T. One enough. Capital T, simple T, heterozygous, heterozygous, capital T, simple T, heterozygous, capital T, simple T, heterozygous alleles. Okay, heterozygous alleles. Okay, so so they so they all are they all are they all are appear they all are appear as told. They all are appear as told. Neither they have capital T, simple T, all of them. So all of them have a dominant one allele. So they are appear told. Okay, all right. So in the bracket, write down all of them have a dominant capital T allele. In the bracket, write down all of F1. All of F1 have capital T dominant allele. right? So one, if there is only one allele, that is enough. If they have only one dominant allele, they can express the dominant feature. Okay, no need having two, neither. So if they have only one dominant, that is enough to express their feature, neither. All right. So that is one enough, right? So F1 generation. Now we have to write down about the, no need to write down in the F2 generations. They are now telling in the number 3, F2 generation cross between two F1 plants. Okay, that means capital T. Then genotypical ratio is genotypical and the phenotypical ratios they are telling. Okay, so we will look at here. I will do at that thing here. Okay, so now I am going to F1 cross with one F1 cross with one F1. So F1 cross with F1. So that means one capital T simple T. I'm going to one capital T simple T cross with one capital T simple T. Now, my dear students, on the ළමයි ටික නේද දක්ෂ ළමයි ටික නේද අත්තරන් ළමයි ටික නේද right. Now tell me, what are the alleles can produce by this parent and this parent? Now I need your answers. So it is 6:45, but we will end this discussions and I will give the interval. So what are the alleles can produce by this parent and this parent? Now they are parents. Now they are parents. So they are coming from the F1 generation. For the F2 generation, then now they are going to produce the F2 generation. Now they are parents. Now these are the parents. Okay. So what are they can produce alleles? Tell me. Yeah. Now I am asking only about the alleles. What are the alleles they can produce? Yes. Here capital T and one simple T. So this plant can produce one capital T allele and one simple T allele. So this parent can produce capital T simple T, and this parent also can produce one capital T and one simple T. So these are the alleles they can produce. Okay, these are the alleles they can produce. So now we have to cross these one parent allele with another parent allele. So first I will cross with capital T with capital T. So it will be give the output capital T, capital T. Okay. So then capital T with the another simple T in the next parent. So capital T, simple T. ඒ කියන්නේ මේ parent ගේ තියෙන හැම allele එකක්ම we should cross with the අනිත් parent ගේ තියෙන හැම allele එකක් එක්කම. So we have to each one allele cross with another two alleles, set of two alleles with the next parent. So capital T is one output, capital T simple T is one output. So all the time, so හැම වෙලේම මතක තියාගන්න capital T එක තමයි capital letter එක තමයි මුලින්ම ලියන්නේ. So that is the dominant one නේ. So capital letter එක තමයි මුලින්ම ලියන්නේ. If you write simple T capital T ඒක වැරදි. හරි? So dominant එක තමයි මුලින්ම ලියන්නේ. So dominant letter එක capital T now you have to follow it first. Okay? So capital T simple T. Right. Then this allele finish. Then go to this allele. So here simple T capital T in a crossing is happen with the simple T capital T, but you have to write down capital T first, then simple T. Okay, that is a rule. Okay, right. Then simple T, simple T. So here both of them are simple, so you have to write down simple T. Now tell me what their feature, what their expression. So capital T mean one dominant allele. ඒ කියන්නේ dominant allele එකක් තිබුණත් දෙකක් තිබුණා. ඒක අනිවාර්යයෙන්ම පෙන්වන්නේ dominant feature එක. නේද? Dominant feature එක පෙන්වන්න so they need only one allele, that is enough. Dominant එක allele එකක් තිබුණොත් එයාලා dominant feature එක පෙන්වනවා. Dominant feature එක පෙන්වන්න බැරි වෙන්නේ මොන වෙලාවෙදි විතරද? Dominant එක allele එකක්වත් නැත්තම්. So here dominant alleles no නේද? No dominant alleles. Only they have recessive values. So recessive values විතරක් තියෙනවා නම් එයාලට dominant feature එක පෙන්වන්න බෑ. So capital T simple T they are short people නේද? So මෙයාලා short and What about the other all? Other all are capital T. එයාලට අනිවාර්යෙන්ම එක capital T එකක් හරි තියෙනවා. So all of them are tall. 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 Okay. So this is the output of F2 generation. So are you all clear? Are you all clear now? Right. How many of you got this idea? Yes. Okay, raise your hands if you got the idea. Okay, well done. Well done, superb. So if you have any doubt, you can write down. But in your tute, you have a blank space in the next part. Okay. So blank space in your next page, in the page number 6, in the top part, you have a space. So you have to write down these all the things in that given space area. Okay. So now I will give the interval. So now 6:50. Okay, 6:49 means 6:15. So come around the 7:10. Okay, 7:10. So please write down these things, then go take the interval. Okay, so I will give 5 minutes to write down, then you can take another 15 minutes as a interval. Okay, right. So we'll see after interval. You can stay here or you can leave and come back. So sometime if you leave, you may have some connection issues. So be here. It is better for you. Okay, right. So we'll see after interval. So within interval, you can share this YouTube link or Zoom link to your any friends. So those who want to study these things, you can share so they can join to the next part of our discussions. Okay, thank you. Okay. How many of you came back? Right. Please raise your hand if you came back. Right. Okay. So sorry for the delay. So we will discuss next part uh, with this with these things. So how many of you written these things in your tute? Do you want any additional time? Okay. So all of you finish neither. Right. Okay. Very good. So this is the Mendel experiment, and this is the monohybrid cross. So monohybrid mean in these experiments we are only check one feature or one inheritance characteristic. So if you consider about the height of the plant, we have two different variation, tall and short. The tall is the dominant one. So we check that tall characteristic feature among this plant. Neither. So P plant we use. So Mendel, these experiments did on this manner. So this is the parental family. You can put P here, then F1 generation, then self-pollination among the cross-pollination, cross among the F1 to F1, then F2 generation. So here. Now tell me, what is the uh, tall to short ratio here in the F2 generation? In the F2 generation, okay, so tall to short ratio. So tall to short ratio 3 into 1. Okay. Tall to short ratio is 3 into 1. So this is the expression. What is the expression from this plant? So what is the expressions they are giving out to the area what we can observe from this plant. So this is the expression. අපිට එළියෙන් බාහිරව දකින්න පුළුවන් externally we can මේ අපිට දකින්න පුළුවන් feature එක තමයි අපි මේකේ කියන්නේ tall the short the කියන එක, right? So it is tall ඒවා තියෙනවා 3ක්, short ඒවා තියෙනවා 1ක්. So it is 3 into 1. Okay. So but we are telling this feature in the inheritance, we are telling phenotype type. So this is the phenotype. So what is the expressions they are given by their output? Expressions tall, 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 and short. These are phenotype. So phenotypical ratio is 3 into 1. So this is the phenotypical ratio that things are given in your tutor. So under the F2 generation, then genotypical ratio is genotype. Gene mean alleles, how their expressions. The alleles, what are the allele combinations? So that ratio will be given the genotypical ratio. So in the here, genotypical ratio, genotypic ratio is what is capital T 1 and capital T simple T 2 and simple T 1. So this is the genotypic ratio and this is the phenotypic ratio. Phenotypic ratio. Okay. So tall and short, right? So these things are included in your tute. So if you want, you can write down in a side. Okay. Right. So this is the monohybrid uh, monohybrid pollination, right? Okay, main monohybrid cross, right? Now, inheritance patterns of monohybrid cross done by the Mendel. Okay, so I will share the screen. So I will share the screen now. You can see the screen. Okay. Okay, so the inheritance patterns of the monohybrid cross, they are given. A result obtained from the Mendel monohybrid cross using 7 different contrasting characters are given below in the table. So in this table is what are the features checked by Mendel. So Mendel did the experiment for uh, this each feature. Okay, so first one is the color of the flower. Okay, so what is the color? He choose two colors. One is the purple one, other one is the white one. Okay, so what is the dominant one? What is the dominant one? Dominant one is the given in the F1 generation. Why? The F1 generation is the dominant one. The expression there. Neither. So purple 705. So dominant one in the F2 generation 705 purple, and white is 224. So ratio is 3:1. So ratio mean the phenotypical ratio is 3:1 on there also, neither, right? So how we can do these type of pollination chart? So now I will give a work for you. So take your notebook or take a paper. So we will go through one by one these experiments by using on this way. Okay, by මේ වගේ අපි කරමු මේ cross pollination ටිකක්. හරි? Right, මේ වගේ මේ monohybrid pollinations ටිකක් කරමු මේ හැම feature එකකටම. Right, okay. So for this every features, for this all features, we are going to do a monohybrid cross. Okay, right. So first, color of the flower, and see about the ratios, and color of the seeds, and yellow and green. So dominant one is the yellow. Then shape of the seeds, round and winkle. Neither. So round is the prominent one here, right? Then shape of the pod. So they are inflated or constricted. So inflated or is the one dominant. Then color of the pod, green or yellow. Then positions of the flower, so axial or terminal. Then height of the plant, tall, short. So this one is already we discussed. Okay, so the tall, height of the plant, this ratio, this experiment we already finished. Okay, so we have to discuss about the next one. I will choose this one. So color of the flower as for a next discussion. Okay, right. So next experiment we are going to do, right? So here two color, one is the purple one, other one is the purple, one is the purple, another one is the white. So now I will give the purple color letter capital P. Okay, so white color allele I will take as a simple P. All P are made. Okay, so this is capital P, this is simple P. Okay, right. So which one is the dominant one? Purple is the dominant. Now take a P1 generation or P1, P or the parental family. Okay, so parent. So what are the parents we are taking? We are taking one purple with cross with one white. So the capital we are taking purple, capital P, capital P1. We are taking white, simple P, simple B. That is a homozygous dominant and homozygous recessive. So this is the homozygous dominant. Then this is homozygous recessive. So what will happen here? So capital P, simple P, simple P. Okay. So now we have to separate their alleles. So what are the separations? Capital P one allele and capital P another allele here. So we have to separate their two parents. Then simple P one allele, simple P one allele. So the simple P, yeah, these are the simple P's. Okay, these are simple P's. So we have to cross them among the generation. So what should be the capital P simple P output, capital P simple P. Then here cross with next gene allele. So capital P simple P. Then with another allele set of allele on the parents. So capital P simple P and capital P simple P. Okay. So this is all are having dominant feature purple allele. So all of them are purple. Now then all of them are, what is the genotype? Genotype and phenotype. So phenotype means what is the expressions they are given to outside. What is the expressions they are given to outside. What is the observable output? That is the phenotype. Phenotype. So here phenotype is purple. Genotype is capital P and simple P. That is the gene expression. So capital P, simple P is the genotype. This is the phenotype. So here purple. Again purple. Again purple. Then again purple. Okay. So all of them are purple. So this is the F1 generation. Then we are going to cross-pollinate. Means again cross among the F1 generation. So F1 generation cross with F1 generation. So I will take one F1, capital P, simple P, then cross with capital P, simple P. So separate the alleles and write down the what are the genotype ratio and phenotype ratio and what can be the F2 generation. So continue this in your notebook. So write down as a question. So write down as a question. Okay. Homozygous purple flower plant. Homozygous purple color plant cross with homozygous white flower plant. Okay. Then cross with F1 give F1 generation. Okay, F1 generation. So question is, what is the genotypic and phenotypic ratio in F2 generation? In F2 generation. So this is the questions. So this is the first step of the answer. So you have to build up another last part of this answer. So can you all do? Okay, this is the challenge for you. Okay, so please do this all things. Right. So the homozygous purple flower plant cross with homozygous white color plants. Okay, then they cross with the F1 generation. So what is the genotypic and the phenotypic ratio? So you know, genotypic is the gene expressions. Okay, gene type. What are the allele type inside the there? Okay, so the allele ratios there. Then phenotypic is what is the expressions they are given in the outside. Okay, right. Yes. Some of them send the answers. Neither. So yes, very good. Your answer is correct. Okay. Others also can send the answers. So because I can see your chat boxes. I can see your chat box. If you made some mistake, I can correct it. Right. Okay. So I will give 2 minutes. Please complete. Right. So this is the first step of the answer. So I take the output of the F2, F1 generation, and I build the F1 generation cross. So you have to build what is the F2 generation, how their appearance are, what is their color of the plants, and the color of the flowers. So you have to build here. So how many of you finished? How many of you finished? Please raise your hands. Okay, good. Okay, very good. Most of you did very good. Well done. So I will go through here. Here, what are the alleles? One capital P alleles and one simple P allele. Then capital P, simple P. Okay. Then we have to cross among the alleles of each individual parents. Capital P, capital P. Then here, capital P, simple P. Then here, capital P, simple P, and capital simple P, simple P. Okay, so output is, what is this? This is purple. This dominant one allele is present here, so it is also purple. Dominant one allele here, so it is a purple. No dominant allele, so it is recessive color white. Okay, so genotypic output. What is the genotypic? Gene expressions. Gene expressions, capital P, 1 into capital P, simple P, 2 into, what is the capital simple P, simple P, simple P, simple P? It is 1. So this is the genotypic. Gene expressions. Okay, genotypic ratio. Okay, then what is the phenotypic ratio? This is the one. So phenotypic ratio is purple into white. So purple 3 and white 1. So this is the phenotypic ratio. Right. So this is the way how we can do the cross pollinations, pollinations among homozygous dominants and homozygous recessives. So this is the monohybrid cross. Why this is called as a monohybrid? Here we are only checking for one feature. We are only checking for one feature. Okay, right. How many of you got this correct answer? All these correct things. How many of you did this all correct things? Please raise your hands. Yes, well done. You all very good. Okay, so you all did superb, excellent work. Right. Now, I will share the screen again. We have to go to the next part of the discussion. So later we come to again this table and we will practice another some features also, how they appear in their generations. So now I will go to the next part of our discussions. Okay, right. So I will share the screen. Okay, so here assignment also they are given here. Analyze the Mendel experiment using the scientific method. So you can do in the later. So select another contrast features from the table and denote dominant and the recessive traits using symbol and create the Punnett square and for a monohybrid. That is the one we are did here. So we did for the color of the flower. So purple and white, how we monohybrid cross perform here. Okay, right. Okay, we will do later for another features. Okay, right. Summary: Mendel experiments demonstrate the traits are inherited in the predictable pattern. So that is predictable, neither. So how it is predictable? If you take a F1 generation, P generation, again P mean the parental generation, as a homozygous dominant and the homozygous recessive, all their F1 is the dominant one. Neither. So all their F1 is represent the dominant feature. But when they come to the after the pollination or the cross among the F1 generation, so they will give the 3:1 ratio phenotypic. Neither. So dominant 3 and recessive 1. So we can predictable that. So that output we can predict. So Mendel create this theory to predict this outcome from generation to generation. Okay, right. Then the study generation. Okay, The study genetic help us to understand how the traits are passed from generation to generation. So this is helped by this understanding of Mendel concepts. Okay, so explanation of pattern of inheritance using probability. Probability means sambhavitavaya. So we can uh, we can find out output of these generations. We can expect the output by using these probabilities. Okay, so first we are going to do a example. So what is the probability we can have tossing a coin? We are going to tossing a coin. That means the coin we are going to put up, and we will find out what is the output from that tossing. So we have two sides. One is the head, other one is the tail. What is the probability of the having tail or the head? So if we perform two times, one time can have head, one time we can have a tail. So the probability is half, half probability here, here, half. Either. So probability is having head is half and having tail is half. So when they are tossing two coins, what are the probabilities are multiple? Okay. Probabilities are multiplied. So when we perform the two tossing, two coins, tossing of two coins. So at the same time, we are going to toss, tossing two coins. So one coin can have a head, other can also having head. So what is the probability for having head? So half. So here half should multiply by another half. So it will be give the output of the final two coins. Okay. So it will be give 1 divided 4. Okay. So this is the when tossing two coins, probabilities head to head. One coin also given head, another coin also given head. This is the probability for having two heads. Okay. So what is the probability for having two tails? So having tail probability is half, having tail probability is half in the each coin. So we have to multiply them. So it will be give the final grand probability of the having one tail in the one coin and another tail in the another coin. So it is also 1 divided 4. Okay, right. So head to tail, head and tail. One coin they have a head and other coin have a tail. So what can the probability? Yes, having head is also half, having tail also half. So it also probability given 1 divided 4. Okay, so these are the probabilities we can take when we are doing perform two times or two coins. Two same feature having we are doing at the moment we are doing two coins, or we can do separately two coins in separate time. Right. So when combining probabilities of the similar outcome, example, head and tail and head. So what we can do, we can take the total of their probabilities. Okay. So when we are combine the probabilities, similar outcomes mean example, head to tail. Okay. One time we having head, another time we having tail. Okay. So what we can do, we can take a sum. So head to tail. Okay, same, right? First time head, second time tail, or first time tail, second time head, like that. So we have a head to tail probability half. Next time head to tail also probability may 1 divided 4. Sorry, 1 divided by 4. So we can may take the sum total. So it will be give half. Okay, so it will be give the half outcome. So this is the probabilities we can expect in the tossing of the coin. Okay, tossing a coin. Okay, so uh, did you got the idea? So do you have any questions? Do you have any questions? Okay, right, right. I think there is a question neither. Okay, clear, ne? Right, very good. So Now I will give an experiment for you, activity for you. There are probability patterns using the beads. Okay, so beads, we are going to beads. We have in the vessels, we have two vessels with the 50 red beads and the 50 white beads in the each. Okay, that means we have a two chambers. We have here 50 red beads and here 50 white. Okay, now what they are telling, procedure, randomly we are going to pick one bead from each vessel and record the color. So at the one time we are going to take one bead out. Okay, so return bead to vessels and repeat 50 times. So we take one bead and check the color and we put into again into the same chamber. Then we repeat that process 50 times. Okay, so counter currents of the red to red, we express as a RR. First time red, second time also red. So red to white, first time red, second time white. Then white to red, first time white, second time uh, red. Then white to white is W and W. So what are the probabilities to having R and R? That means first time red and second time red. What is the probabilities? Now fill this table with your knowledge. So what is the probability having red in the first time? What is the probability having the red in the first time? It is the half. It can be the red or it can be the white. Neither. So here also probability 50. So not in the white though. Here also white 50 and uh, 50 red. Okay, both of them have 50 white and 50 red. Okay, that is the matter. So if you take one coin, it can be half. It can be half probability to take in the white or red out. Neither. Right. So now what are the probabilities? Tell me. What are the probabilities to having R and R? So we have to take initially first. If we take the first red, the probability is half. Neither. And next time also red, we have to multiply by half. So the grand probability is 1 divided 4. So grand probability 1 divided 4. Okay. Then what is the probability of the red and white? Red and white. So red probability half, white probability also half, multiple by it also give rise the 1 divided 4. White and red. So initially white half, then another red also half. So this is given the output of 1 divided 4. Then WW, that also white and white. So half multiplied by half. So here we can take the probabilities of, yes, 1 divided 4. So all of them given the 1 divided 4. What is the combined RW? That means the red and white and red, same outcomes. So what can be the same outcome? If we check for the same outcome, we can take the sum. Okay, so we can take the sum. So RW, the ratio probability is 1 divided 4. WR probability is 1 divided 4. Now we can take 1 divided 4 plus 1 divided 4. So the probability, grand probability 2 divided 4. Okay, that means we can tell it is equal half. Either 2 divided 4 means that is equal half. Okay, right. So ratio is the ratio. So what the ratio they are asking? A ratio, we have to predict the ratio among R to R ratio. So that is not printed here. RW, sorry, having RW and OWR, then WW. What is the probabilities? Ratios. So if you take same time, first one should be the R and second one should be the R. The probability is now ratio. Now it is coming as a ratio. It is 1 into 2 into again 1. Okay. So this is the ratios we can take in this experiment. So this, okay. So are you clear? Are you clear? Or do you have any questions? Okay, so probabilities, these things are you are learning in the mathematics also, nene. So these are simple for you. So you can easily put these probabilities and you can put the ratios. So in the science, this is using in this inheritance part. Okay, right. So uh, in the medical science also we are using in our hospital setups, we are using statistic. This we are calculating these probabilities by using mathematics. So we have some probability charts. We are using some methods. So if you have a patient, the patients have a smoker. Patient is a smoker. So he has a risk. He has a risk of developing cancers. So if the patient has a smoking history, and what is the probabilities to having the cancers? So we are calculating how long they smoke. So what is the connection between smoking and the lung cancers? So these are the probabilities we are calculating in hospital setup in the medical professions, but in your level that is are not in your syllabus. So this is one type of probabilities. So you can take the probabilities and you can multiply by to take the repeated time. Then if you take a combine, you have to take the sum. So then you can convert them into the ratio. The ratios will be given like this. Okay, right, fill this all these spaces. Now what is the purpose is probabilities? Learning this probability, what is the purpose? Purpose is when we are going to perform a Mendel monohybrid cross. Okay, when we are going to perform a Mendel's monohybrid cross, these pollinations, may probabilities will help to determine the output. Okay, or it will be help to predict the outcome. It will be help to predict the outcome of the monohybrid cross. Examples they are given here. So in this example, so write down, I will give a little bit detailed example. Okay, so if the parental, if the parental uh, parental plants having if the parental plants having dominant, oh sorry, homozygous dominant and homozygous recessive alleles, homozygous recessive alleles. Okay, we can predict their F1 their F1 generation. All plants should be dominant plants. Dominance for dominant plants. Okay, so this is one example. Okay, this is one example. If the parents are homozygous dominant and homozygous recessive. What their F1 generation, all their plants are, all their outcomes are dominance. This is our prediction. So this can be predicted by ourselves. Neither. So Mendel tell how to predict this. Neither. Then if the second one, okay, second example, if the parental plants heterozygous, okay, heterozygous, their F1 generation or their next year, their F1 or F2 F1 generation or their next generation, okay, F1 generation outcomes 3:1 phenotypic ratio, okay. If the parent plants have heterozygous in their heterozygous, their comma and their F1 generation outcomes 3:1. Okay, so I will explain that in the whiteboard. Okay, so don't worry about that. I will explain that. Okay, right. So now see in the whiteboard, I will explain. So if your parental plants, okay, if your parental plants, if they heterozygous, what will happen? Now I told their phenotypic ratio 3:1. So I will give capital T simple T and capital T simple T. So this is also heterozygous, no, heterozygous, different alleles. Here heterozygous. Heterozygous means different allele. Homozygous means same alleles. So what are the alleles can present here? Capital T one allele and simple T one allele. Then here capital T one allele, simple T one allele. Then we are cross here. So capital T, cross here. Capital T, simple T. So here finished. Now this gene. So this allele combination. Now capital T, simple T and simple T. So these three are dominant feature. They have dominant feature because of they are having dominant alleles. This is the recessive feature. So recessive feature because of they are having only recessive genes. Okay. So this ratio now, what is the ratio? 3:1. So the phenotypic ratio or the observable feature of the ratio is 3:1. So we can predict this neither. So by the way, when you are going to the A level, so don't forget these things. So if you are wish to do biological science, so you have to study biology. In the biology, you have to calculate and you have to predict next generation, what will happen to next generation, what are the ratios. So you have to predict there. So this is the fundamental basic foundation for that A-level subjects. Neither. So don't forget these things. If you have any doubt, please clear that doubt at the same time. Don't get that doubt into the until in the exams. Neither. If you are go with the doubt, you will definitely struggle in the exams. Okay, right. Right. So this is the way how we can predict neither. So 3:1. Now my prediction is correct. Right. No need to write down these things. So we will move on to the next part. So next part is the basic concepts. What we are learning here, we have to discuss with the summaries. Okay. Right. Now I will share the screen again. Okay. Okay. Are you all hungry? Neither. But again, right. So basic concepts. Basic concept. First one is the genes and inheritance. So what now we are going to discuss about what is the connection between genes and this uh, inheritance. Okay, so Mendel identified that an organism feature are determined by a specific factor. So what is this specific factor? Later named as genes. Okay, right. So Mendel find out what is the factor is determine these features. The feature is determine this fact. All inheritance are genes. Okay, then gene representation. He represent dominant traits. We know now how we can gene representations by the dominant way, right? Okay, so dominant traits, right? So dominant traits, how we can use capital letters. So these are the basic concept we already know. Neither. So dominant traits we are using capital letters. Example, if the R for round. Neither. So other examples, tall we represent by capital T. Neither. Right. So recessive traits, how we can do lowercase letter or the simple letters. So examples they are given simple R for the sprinkle seeds. So another examples we can provide here, short plant, we are using simple T. If the tall one capital T, definitely the simple letter should be the recessive one. Neither. Right. That recessive is short, right? Gene combination. So homozygous identical alleles. All right, homozygous identical alleles. Examples we can provide here. Capital T, capital T. This is dominant feature. Capital T is capital T. Then simple T, simple T. This is dominant one. This is recessive one. Okay. Right. So what about the heterozygous? Different alleles. We know these things. So examples, if we take capital T, other one should be simple T. Neither. So these are the examples for the heterozygous different alleles. Okay. Gene expressions. Okay. Gene expressions. So the combinations of the gene pair for a particular traits, okay, traits in the organisms, gene is known as gene expression. So combination of the gene pair for a particular traits. So example, R, simple R and capital R. That is a gene expression. Gene expression of for a particular feature. Neither. So this is simple. If we take R, if we take capital R as round seeds, okay, simple R as a wrinkle, yes, wrinkle seeds. Okay, so what is the gene expression here? Capital R, simple R mean round one gene and wrinkle one gene, one allele. So both allele combined together give a gene expression. Okay, so this is another gene expression, RR. So both alleles are represent the dominant neither allele feature. Neither. Then simple R. This is another expression. So another expressions we can tell here, another examples you can write down. Capital T, capital T. This is one expression. So what is the gene expression here? They are telling they are both alleles are dominant for that feature. Neither. So both allele are dominant for the feature. Okay. Then capital T, simple T. What is the expression here? Gene expression. One allele is dominant allele, another one allele is a recessive allele. Okay. So one is dominant and one is recessive. Okay, right? So that is the examples we can provide here. Then we have to write down the phenotype and the genotype. So we will go to the phenotype and genotype. So write down with me what is the phenotype, what is the genotype. So we know what is the phenotype. Pheno mean external appearance of the traits, right? So write down here external appearance of external appearance of traits. Okay, external appearance of traits. Okay, that is the phenotype. Phenotype mean in the Sinhala අපිට බාහිරින් පේන්න තියෙන මොන ලක්ෂණයද? That is phenotype. Okay, අපිට බාහිරින් පෙනෙන දේ phenotype. නේ? What we can observe from the outside, that is the phenotype. නේද? In Tamil வெளியிருந்து பார்த்தா என்ன விளங்குது? That is the phenotype. නේද? So in outside surrounding, what we can observe, that is phenotype. Okay, so genotype mean the gene expression. Gene expression mean what are the genes inside their traits. In the traits, what are the genes inside? So if they have two genes, both of them are dominant, or both of them are recessive, or one dominant, one recessive, that is represented by their genotype. Okay, that is the genotype. Now you can take the idea, right? Now genotype, right? We will write down gene, write down here gene composition, for that traits. Okay, gene composition for the traits. So that is we call as a genotype. Okay, right. Examples, we have a genotype, simple R, that is the homozygous genotype. Okay, simple R, capital R, that is the heterozygous genotype. These all are typically phenotype type for the round seeds, and the we are taking for round seeds. Round seeds. So both of them are round. Capital R is the round. So they are round. So this R are also a round phenotype. This simple R also round phenotype. Pheno mean expression. What is the external feature? But the inside the gene, they have one is homozygous status. They have capital R. In the heterozygous status, they have simple R. All right. Okay. Now, nature of the genetic materials. So before going to this part, I would like to go for another question. So I already told you we have to practice the questions here. So I will go to the again into this table. So Mendel experiment, I will give here. So you have to predict the outcomes. Okay, right. Now I will give a little bit different manner. So I will give different outcomes with different way. Okay, so we will check for the another way. So we will check for the shape of the seeds. We will now we are going to do the experiment to find out these ratios of the shape of the seeds. Okay, right. Okay, I will give the questions in the whiteboard. You can follow me in whiteboard, right? Okay, now the question is, okay, homo, write down with me in your paper or your book, text me writing book, that is okay. Homozygous round seeds plant, okay, cross with homozygous wrinkled seed plant, wrinkled seeds plant. Okay, in the F1 Then, okay, then F1 generation plants cross among them and get the outcomes of get the F2 generation, get the F2 generation. Okay, now first question is, okay, first question is, what are the genotypic ratio in F2? Okay, the second question is, what are the phenotypic ratio in F2? Okay, so you can take the alleles capital R as a round. So you can take the allele capital R as a round, simple R as a wrinkled. Okay, so these are the allele representations you can use. So I am not going to help more. So this is the questions. I will give you 5 minutes. So try these questions, get all the outcomes with the previous manner. නේද? කලින් කරා වගේම අඳින්න හැම එකක්ම if so you have to put T, what is the parental genes, then you have to predict the F1 generation, then you have to cross among the F1, then you have to find out the what is the F2 generation. So these things you have to do here, then you have to individually find out the genotypic ratio and the phenotypic ratio. Okay, are you all clear? Okay, so I will give 5 minutes. You can do within 5 minutes. I will sit here for a while until you are doing. Okay, right. If you are finished, please raise your hands. Then I will come to the discussion. How many of you finished? How many of you finished? Please raise your hands. Yes, one was okay. One finished, neither. Yes. What about others? So this is the questions. You can read. Homozygous round seed plant cross with the homozygous wrinkled seed plant. Then F1 generation plants cross among them and get the F2 generation. So what is the genotypic ratio in F2 and what is the phenotypic ratio in F2? Yes. Next one person finished. Yes. Okay. 2 people. 3. Okay, very good. Yeah. Okay, 4. Yeah, all are very good. Super. ආස හිතනවා ළමයි උගන්නන්න ඇත්තටම නියමයි. මේ එකක් මම මේ වැඩත් එක්ක ඇත්තටම හොස්පිටල් එකේ ට්‍රේනින් වැඩ so මම තාම ට්‍රේනින් ඩොක්ටර් කෙනෙක් නේ ඔයාලා දන්නවා ඇති මම මගේ තාම ට්‍රේනින් පීරියඩ් එකේ ඉන්නේ so I am doing my training at the hospital නේද then I am very මම ගොඩක් ආසයි මේ උගන්නන්න නේද මේ I very love to teach so ගොඩක් ආස හින්දා මම part time උගන්නනවා එතකොට I am a teacher as well නේද so I am doing two subject teaching so that is very complex subjects one is the A level chemistry Another one is the O-level English medium science. So A-level chemistry ළමයින්ට උගන්වන ගමන් හම්බෙන part time වෙලාව ඇතුළත prepare වෙලා ඔක්කොම tutes හදාගෙන නේද? ඔක්කොම මේ වගේ tutes වලින් හදලා ඔයාලට mind maps හදලා ඔක්කොම කරලා උගන්වන්නේ මගේ ඇතුලේ තියෙන ආසාව. ඒ ආසාව වැඩි වෙනවා අල මේ වගේ හොඳට perform කරද්දී. So I love to teach නේද? I love to teach more and more. So ඔයාලා මෙහෙම perform කරද්දී I'm very happy. So thank you for your performance නේද? So I expect all of your uh, output නේද? You all should get A pass in the science නේද? That will be give the happiness for me. So I I wish ඔයාලා හැමෝටම ගන්න පුළුවන් වෙන්න කියලා මම ප්‍රාර්ථනා කරනවා. So අපි යමු වැඩේට. Okay. මෙහෙම මේ ඔයාලා ඉගෙන ගන්නවා දකිද්දි සමහරක් දවස්වලට මම පාන්දර 3-4 වෙනකල් ටියුට් හදනවා. ඔයාලගේ මයින්ඩ් මැප්ස් හදනවා. ඒකම කාටවත් දෙන්නේ නැහැ මැටීරියල්ස් හදන්න. මොකද my knowledge should be included in this tute. So my tute, all the tute materials are done by my hand. නේද? මම හදන්නේ, මම edit කරන්නේ, මම type කරන්නේ. ඒවා මම කාටවත් දෙන්නේ නැහැ කරන්න because මේකේ පොඩි mistake එකක් වුණොත් this is your life. නේද? ඒකින්ද this all responsible will take with myself. Then I am doing all the works until early morning නේද? Sometime I'm not sleeping well, but I will do these things for you all. ඔයාලා ඉගෙන ගන්නවා දකින්න ආස හින්දා මේ හැමදේම කරනවා නේද? ඒකින්ද ආසාවෙන් කරනවා. Right? Okay. Good. So very happy. Right. So P1 generation මගේ කතාවම වැඩි කියන්නේ නැහැ තැන් වල මම මහන්සි වෙනවා වැඩි කියන්නේ ඉතින් කියන්න හිතුණා ඉච්චරයි. Okay. So capital R cross with simple R. So what are the genes here? Capital R 2 genes, alleles 2 alleles, simple R 2 alleles, then cross among them. So cross among them, capital R simple R, capital R simple R, and capital R simple R. Here capital R simple R. So all of them are round seeds. All of them are round seeds. Now F1 cross with F1. So capital R simple R cross with capital R simple R. Now capital R one gene, one allele, then another capital R and simple R allele set of alleles. Now we cross with them capital R, then capital R simple R, then capital R simple R, then simple R. Now what is the output here? So output is genotypic ratio. Genotypic ratio is capital R to simple R, capital R to simple R to simple R is 1 into 2 into 1. So that is the ratio. Right. Then phenotypic ratio is round seeds with the wrinkle sheet. So how many around here? All dominant are round. So 3. So winkle is 1. So you can do neither. Right. Okay. You can do like that. Any Mendel experiments you can do like in this manner. Right. Okay. Right. Now we will move on to the next part. I will share the screen and I will do these things in iPad. Okay. Okay, I think you no need another interval neither. So I gave the little bit long interval. So we'll see. If you want, I can give 5 minutes around. Oh, what is the time? Oh, 8:17. So I will give 5 minutes around 8:50. 8:50 wage veddi. So I will give right. Let me share the screen. Okay. Now we will move on to the next part. So next part is nature of the genetic materials. Okay. So we'll see about the nature of the genetic materials. The genetic materials is the deoxyribonucleic acid in the chromosomes. Okay. So we know we have nucleic acid. So nucleic acids are made by deoxyribonucleotide. So combinedly they have formed the nucleic acid, one type of nucleic acid, DNA, another type of nucleic acid is RNA. So we have a DNA in the human bodies. Okay, so deoxyribonucleic acid, that is called as a DNA. Okay, in the chromosomes. So discovered this was Watson and Crick in the 1953. DNA has a double helix structure. So Watson and Crick introduced the double helix structure in the 1953. Then DNA nitrogenous bases adenine, thymine, cytosine, and guanine. Okay, so base pair rules pair with the adenine pair with the thymine, cytosine pair with the guanine. Okay, so that is the way how we are alleles are combined. Okay, right, now we have to draw something here, so I will draw in the digital screen so you can copyright them. So in our chromosomes, okay, so I will take a pair of chromosomes. So these chromosomes, one chromosomes are not pair. So chromosome, what we have inside, we have DNA. Okay, so DNA having in the chromosomes. Okay, so that DNA are double helix. Double helix mean there one DNA chain is combined with the another DNA chain. Okay, so this is the double helix DNA chain. Okay, double helix DNA. So in this double helix DNA, what are the things we are having? In some areas we are having what? We are having nitrogen base pair. Okay, in these genes we have uh, adenine, and other side there should be, so I will zoom area. Okay, so I will zoom one area, sorry, then I will draw again the zoom area. Okay, so I will again draw this zoom area. Okay, all right, because that is very small ne? So I will draw this zoom area now. So in this zoom area we can see we should have adenine, and opposite side we should have thymine. Okay, so interconnection should be happen in between this adenine and thymine. So with the reduce the thickness of the pen. So adenine and thymine. Okay, so they are interconnected together. Then if you have a cytosine, it should be interconnected with opposite side guanine. Okay, so again, if we have cytosine, it should be guanine. Neither. So opposite manner also we can have. Neither. If you have thymine here, other side you should have adenine. Okay, so this is the way how these nitrogenous bases are going to combine. So please draw these things in here. Okay, please draw this. Okay, so in your A level, you will learn these things more deep and more complex manner. So if you enter to the medical schools, you will learn more and more complex theories. Okay, right. Okay, so please draw these all the things. Then we can come to the next part. Okay. Okay, right. So chromosomes, they are having inside the chromosomes, we are having DNA. Inside the DNA, what we are having? Nitrogenous bases combined together, right? So nitrogenous bases are combined in a way, adenine pair with the thymine and cytosine pair with guanine. Okay, so that combinations are available throughout this DNA chains. Okay, right. Now, what is the gene now? What is the gene? The gene is a specific DNA segment coding for a particular trait. So, if you consider this DNA part having this DNA part responsible for the some particular traits. If they're responsible for some particular traits, we will call them as a gene. So gene is a part of the DNA. So we can tell this part as a gene. Okay, so they are responsible for some traits, some features. So they are going to uh, they are going to express some features by this nitrogenous base segment arrangement in the DNA. So this area we call as a gene. So now you can tell a gene is a specific DNA segment coding for a particular traits. Okay, genes determine the features by the sequence of base pair in the DNA. Right, now you can see gene determine the features by the sequence of the base pairing in the DNA. So we can draw here as well. So I will draw simple thing. So in this manner, we can tell. So here I will put adenine, thymine, okay, adenine, thymine, cytosine, guanine, then again adenine. So what are the opposite one? Adenine should combine with thymine. So thymine should be opposite here. Adenine is opposite here in front of the thymine. So cytosine now guanine, opposite side guanine. Guanine now cytosine in the opposite side. So adenine again thymine, they are opposite side. They should be have thymine. So this segment is specific for a one trait. Okay, that is called as a gene. Now that area we can call as a gene. Okay, so that area we can call as a gene. Right. So you can draw these things here. Okay, so please draw these things. Okay. This is not the simple usual seminar. So this seminar will cover all around this topic. Neither. So I like to teach all the things. Neither. All the things regarding this lesson. Right. Then we are going to the gene linkage. Okay. Gene linkage. So gene linkage, it is the homozygous chromosomes carry gene for the same traits. Okay, so homozygous chromosomes carry gene for the same traits. During the gamete formation, gene may segregate independently as shown by Mendel. Okay, so homozygous chromosomes, they are carrying the same genes for same traits. Okay, during the gamete formation, genes may segregate independently as shown by Mendel. Okay, so when they examples, if they have, okay, if we have two chromosomes, if I take two chromosomes, this is two chromosome. In these two chromosomes, they have some area, no? So some area, I will take this area as a gene. Okay, so in this gene area, if I have a capital T here, sometime I will have a simple T here. Either. So they will segregate when they are gamete formation. Now these 2G may chromosomes are combined together. Then when they are formed the genes, when they are formed the gametes, these two chromosomes are separate. So these two genes will be separated. So that will be called as a gene linkage. So here the genes also linkage. Okay, so you can draw here these things. Okay, what will happen? These are during gametes formation. Genes will segregate or they are separate. Okay, right. So you can draw these things in the given space. Right. So please write down those things. Okay. Then next part, linked genes. Linked genes means they are linked. These discovers by the Morgan are present at the same chromosome and do not segregate independently. They sometimes their linked genes are they are present in the same chromosomes. Okay, they are present in the same chromosomes. They are not going to separate or segregate independently while they are forming the gametes. Okay, so these genes are called as a linked genes. Okay, so these are the linked genes. So if they are linked genes are not going to separated, but the independent alleles or independent genes in the both two chromosomes, if they form in the gametes, they will segregate or they will separate during the gamete formation. Okay, right. That the example is, you can put the example, if the maternal or if the mother two chromosomes, if the mother has two chromosomes, okay, so mother has two chromosomes, okay, each chromosomes, one chromosomes have the capital T and one chromosome have a simple T. Okay, right, I will draw clearly. This is mother chromosome. Okay, in the mother chromosomes, she has capital T in one allele, one chromosome, simple T in one chromosome. Okay, when they form gametes or when she form a ovum, she produce one capital T chromosomes to one child or one ovum, or and she will give the simple T into the another ovum. So they will separate independently. Right. So if the father has uh, if the father has two chromosomes, one he has R, capital R and simple R gene. The simple R and capital R responsible for the one specific feature kiyamuko. So if they form the sperm, if the father form the sperm, so one capital R gene separated for one sperm and another simple R gene is separated for the one sperm. So what type of the sperms going to what combine with the what type of the ovum, that will be determine the outcome of the their next generation, right? So that is the gene linkage and gene about the gene, right? Right. Now assignment also given here. You can read about the assignment and you can do it your own. Then Mendel's experiment and demonstrate that traits are inherited in the predictable pattern. We know about these things we discussed. Then the study of the genetic helpful to understand the how traits pass from the generation to generation. So this is important to find out these things. Then hereditary of humans. Now we will move on to the hereditary of humans. Okay, right. Now, what are the hereditary? What are the hereditary? So we will see hereditary is the transmissions of the inherited characteristic from one generation to next generation. So hereditary means simple term we know. One characteristic feature transform from the one generation to next generations. We are calling as a what is Yes, hereditary. Okay. So heredity, right? So the process which the process by which these characteristics are transmitted is known as inheritance. Yeah. Now you can see the term. What is the term they are trying to give the I? What is the main? Sorry. Wait a minute. Yeah. Okay. My pen is going battery low. Right. Okay. Now I charge the pen. So these explanations give the idea about this, doesn't it? So heredity means transform the characteristic feature from one generation to next generation. This process is called as a inheritance. This process is simply we can call as a inheritance. Neither. So inheritance is a important process. Neither. So it will be how it will be tell how the characteristic inheritance features or traits go from one generation to next generation. That is called as a heredity, right? So concepts, key concepts we should know about this: gene and chromosomes. So characteristics are passed to the generations through gene located on the chromosome. So we know about that, right? So genes and the chromosomes. Characteristic features are the features are passed from one generation to next generation through the gene located in the chromosomes. We know neither. So how it is happening. Then gene linkage and meiosis. So behavior genes and chromosomes during the inheritance explained by the gene linkage and meiosis. So do you know about that? When they have two set of gene, two set of chromosomes, they are going to separate. Before the separations, their same area genes will be going to linkage and they are separate their material with them. Then only they will go in the, with the meiosis, they will go into the separation. So with the meiosis, they will separate into two. Okay, right. So, not the meiosis first one they are going to not separate. In the second meiosis they are going to separate. Okay, right. So these are the things we should know here, right? Okay, so you all know about the meiosis and how it is happening, so you can link those knowledge in here, right? So meiosis, when they are produced in the meiosis is important to produce the gametes, no? So when they are produce the gametes, this linked chromosomes inheritance will be uh, separated. Okay, right. So number of the chromosomes for a specific feature is a constant number. Okay, so a specific feature has a constant number of the chromosomes. So if you tell pineapple, if you know pineapple, they have 50 chromosomes. So garden pea, they have 40. So onion 60. Neither. So maize 20, paddy 24, tomato 24. So this value you can roughly memorize, but not compulsory. Yeah? So organisms numbers may organism host, the number of chromosomes is 33, but the mouse 40. The human, how much? 46 chromosomes and 23 pairs. 46 individual chromosomes, 46. If you take them as a pair, we have 23 pairs. Okay, right. So these are the things we should know under the this heredity and inheritance. Okay, right. Now we have a next chapter, sex determination. Okay, right. So I will go to the sex determination. I will go to the whiteboard. Okay, so what is happening when you going to, when to one man and one female, now they are got married. So now they are going to produce a baby. So the man give the sperm Man give the sperm, girl or female give the ovum. Okay, so they are going to fertilize. Okay, they will produce the zygote. They will produce the zygote. Okay, they will produce the zygote. So in this sperm, the man have some chromosomes, specific sex chromosomes. The sperms will carry that sex chromosome, and female have the specific sex chromosomes. So that female ovum carry that sex chromosome. Then they will fuse together and form the zygote. Okay, so the chromosomes or the what are the sex chromosomes present in the sperm and what are the sex chromosomes present in the ovum will determine the sex of this baby. Okay, so if the baby want to be boy or if the baby want to be girl, it will be determined by the chromosomes in the sperm and the ovum. Okay, what are the usual chromosomes present in the sex chromosome present in the male? Male, they have a sex chromosomes X and Y. So if they have X and Y, we call as a male. Okay, so female, they have only XX. So these are the sex chromosome. So if this sperm is X, if this now tell me what type of the genes produced by the male? Male can produce X or Y, but definitely the female, she can only produce X gene. So definitely this ovum carry the X gene, but the sperm can carry X. If they both of them, the zygote will be XX. So this baby will be female. Okay. So if the sperm carry the capital Y or the Y gene, so this baby will be XY. So this will be baby boy. So the sex determination will be depend on the sperm specific, especially in the sperm. Why? The sperm has two different sex chromosomes. One is the X, another one is the Y. But the female, they have X and X same chromosome. So how all the time, neither every time the female, the girl or ovum, their ovum carry the X gene, X. But the male sperm, they have a variety. They can bring X or Y separately. Neither. So if they carry X, their output can be the girl. If they carry Y, the output can be the boy. So these things we have to write down. Okay. Right. We'll move on to the note. Okay. So these things are very easy things. So we will write down here. Share the notes. So are you all tired? Mahansida? Okay. Please may chat with me. Otherwise, I am also boring. නේද? පොඩි chat එකක් දාන්න. නැත්නම් මට නිින්දයි. නේද? Because no one in front of the area නේ. So you all are at home. So I am also feeling tired and feeling sleepy. Right. Okay. Just for a joke. Yeah? Right. So මොන හරි chat කරන්න. Right. At least ඕපදූප හරි අහන්න. අහන්න දෙයක් නැත්තම්. නේද? If you have nothing to ask, මොන හරි ඕපදූපයක් හරි අහන්න නේද? I will tell you නේද? So I have so many stories. But if you ask only, I will tell you. නේද? Stories ගොඩක් තියෙනවා. ඇහුවොත් විතරයි කියන්නේ. So if you follow my TikTok channel, you can find out so many stories I upload there. නේද? Right. Okay, that is me. Separate part. Now we are going to sex determination. Okay. The sex determination is the process. Okay. This process which an individual gender is established, a human has 46 chromosomes, okay, and 20 or 23 pair. So out of these 22 are autosomal and Remaining one pair is the sex chromosome. Remaining one pair is the sex chromosome. So that pair is determine the sex. Okay, right now female sex chromosomes. So what are the female sex chromosomes? Female have, okay, we will write down. Female have two X chromosomes, or I will write by the word two X chromosomes. Another 2X sex chromosome. Okay, so XX, they are XX. So what is about the male? Male, they have one Y and one X chromosome. So male are XY, right? So male are XY. Then, okay, additionally, you can write down Y chromosome. Y chromosome is smaller than X chromosome. So this is additional feature. Neither. So Y chromosome is smaller than the X chromosomes. Okay, right. When did you become a doctor? So qualified doctor, my training will be finished within next 1 year duration. So I have to train another 1 year duration. So most probably this is 2025. Ne? So if they have no any strikes, no any other delay, so I will become a doctor in a 2026. Okay, so 2026 is the my appointment. I will get my appointment in 2026, right? Okay. Right. Okay. Then formation of the gametes. How formation of the gametes will be happen? Now I already explained. So one chromosomes from the one sex chromosome from the sperm and one sex chromosome from the ovum. Okay. Right. So we will write down during the formation of the egg and during the formation of the egg and sperms, the pair of sex chromosomes, the pair of sex chromosome, pair of sex chromosomes, okay, pair of sex chromosomes separate. Okay, so egg, how they having, what they having? They having always having X chromosome. Okay, the sperm, what they are having? The sperms, they are having Y chromosomes or X chromosomes. So they can having Y chromosome or X chromosome, right? They are having X chromosomes or Y chromosomes, right? Then fertilization, right? We go into the fertilizations. We know these things. So under the reproductions unit also, I told these things. So if you are not participate to that seminar, you can go to the YouTube, type on the search bar, Science with Dr. Sir. You can find out our YouTube channel. Then go to the seminar video of the reproduction, man reproduction part we discussed in last year, neither, right? Okay, and other seminar video also available. Rate of reaction seminar also available there. Some paper discussions also available in our YouTube channel. So if you want, you go and see that video in our YouTube channel. Okay, right. So now fertilization and gender determination. So zygote formation, when the egg, egg it carries the eggs fertilized by the sperm, result in zygote can be have. Yes, what can be have? Yes, result they can have XX or XY output. Okay, so egg X fertilize the sperm. Okay, if the sperm carry Y chromosome, if the sperm carry Y chromosomes, we can have XY. So this is baby boy. Baby boy. Neither. So if the sperm carry X chromosomes, it will be XX. So baby girl. Baby girl. Okay, right. Then we can on, if you want, we can write down here. Please the draw. This is the sperm. So this is the ovum or the egg. This is the egg. Then they will combine together, form a zygote. Okay, this is from the zygote. So sperms can carry what? So sperm carry X or Y. So egg carry X. So zygote can be XX or XY. So you can draw these things in this given space. Okay, so complete this note like this. Okay, are you all clear? Are you all clear? Right, very good, very good, very good. Clear. I have another set of questions. Yes, and not a questions. Answer. Answer දෙන්න කියලා හිතාගෙන open කරේ. There's not questions නේද? Right. Okay, so finish these things. Then we will, the probability, the chances of having girl or boy, 50-50. නේද? If the baby boy, if the father carry the Y chromosome, the output is the baby boy. If the father carry the X chromosome in his sperm, what will happen? The baby girl output. Sometimes you may realize some families they having only girls, neither no any boys in that family. That is due to his father's sperm only carrying more prominently X chromosomes. That's why their family having only girls, neither no any boy member, neither, right? So some families they having only boys, neither. So that is due to his father's sperm, their father's sperms, they are prominently carry the Y chromosomes unless the X chromosomes, neither. So Y is prominent there, so they will produce the, yes, what is, they are prominent X baby boys. Okay, right. Illustrate the process of sex determination in the human. So I'm going to illustrate in the here. Okay, I will illustrate in the digital pad, and I'm going to lazy to go to the board. Okay, right. So if the sperm, okay, if the mother, if the father's sperm, okay, I will show something in your textbooks. So Okay, so can you see my screen? Okay, is there any disturbances? No, ne? Okay, so second, so we can in the diagrammatic manner. Oh yes, so we can have, right. So I will go in the diagrammatic manner. So one X chromosomes and another X chromosome, this is present in the egg. This is present in the egg. So it will be crossed with the, what is the sperm. So a sperm can be have X or Y. Okay. So a sperm can be have a X or Y. Okay. So the X is large, Y is shorter. Right. So what will happen? Now they are going to produce the sperm. Okay. So this is the, this is not the egg cell. This is the egg cell division. Cell divisions happen using this egg cell. So if they have X chromosomes, okay, now here also X chromosome. Then here the sperm productions, when the one sperm can X and other sperm can be Y. This is Y. So now we have to combine with this parent. So here what is happen? Here X with X. So this is XX girl. Okay. So here one X and small y. So this is XY, that is boy. And again here X, X, this is girl. And here then XY, so that is uh, XX girl, here XY boy. Okay, so this is the way how we can determine the sex. So I go through your textbook. So 50-50 chances is available here, but you can separately in the easy manner, easily, not going through this complex diagram. So easily you can using this diagram. So I will write down in the red color. So red color diagram, you can tell XX and XY. So it will be separate into 1x, 1x, then here 1x and 1y. So we can cross them, x, x, here x, y, then here x, x, and here x, y. So here this girl, boy, girl, boy. So ratio is girls 50%, boys output 50%. So you can write down these things in a given space. So these both two manners are correct. Neither both two are corrected. Okay. Do you have any questions? Ah, yes. Insert siblings in other. Yes, I have one brother. Eh, yes, one brother. Very elder brother. But what I got a look. Right. Do you have any questions? Lesson के questions you can ask them. Yeah, good question. Sir, marry කරලාද? Yes, recently married. තාම දවස් දවස් ගාන මතක නෑ. So last December 21st මේ මැරි කරේ. හරිනේ. තාම මාසයක් වෙලත් නෑ. තාම දවස් 15 විතරයි. හරි. මැරි කරලා. right නේ. The recently married. Okay. මේ she also a doctor. So works in Kurunegala Hospital. So I'm working Batticlo Teaching Hospital. I am training in Batticlo Teaching Hospital. She is training in Kurunegala Teaching Hospital. So okay. So both of them are in separate two places. Yeah, there is a very nice love story in my university's life. So I will tell in the next class. So ඒ වඩක් නෑ ක්ලාස් එකේදී නේද? බෑ දැන් කියන්න බෑ. No time නේ. So ඊළඟ seminar එකේදී කියන්නම්. So okay. Okay, right. So you finished that. Ne? So now these are the some autosomal chromosomes and sex chromosomes appearance. Okay. So may this may representation is may gene study. Okay. We are going to gene study. So we are going to study every pair of genes in the body. So we have 23 pair. No? So we going to 23 pairs of gene. So when we study 23 pairs, those 22, all these 22s are autosomal. And one pair, it is the sex chromosome. Now look at that sex chromosomes. This is the large one. This is the small one. So large one is the X, small one is the Y. So this is the boy. This is the boy. Okay. So this diagram is showing the boy. So if you go to the right side diagram, so all these pairs, all these 24 pairs. No. Make a pair make a zoom කරලා තියෙන එක තියෙන්නේ. So 22 pair 22 23 and what is this 24? So bloody idiot. So this is wrong, eh? So wrong diagram එකක් මේක. So මේක 24 කියන එක නෑ. Okay. Okay, they have only 22 autosomal. මේ ටික cut කරගන්න. So diagram එක wrong. So this is autosomal and this is sex chromosome. In the sex chromosomes, they have both X and X. So this is the girl. Okay. So this is the girl. Okay. Right. Now we have to discuss about the human inherited disorders. Okay. Now I think you all may feel tired. Neither. How many of you feel tired? Please raise your hand. How many of you feel tired? Yeah. What is my name? A good question. Neither. So I told me, please find out. Neither. That is your duty. So find out my real name. Okay. Right. So how many of you feel tired? You all are not feeling tired, ne? Okay. But I am feeling tired. Oh, no tired. Okay. My age. Good question. So I am 26, 26-year-old. Okay. So born in 1998. Right. Birthday, that is your duty to find out. Birthday most probably coming. No, that is your duty. Right? So you can find out. So, right. So may you can find out me by this way. So take a picture of myself. You having in Tute or in the group my profile picture in the WhatsApp. So take that picture and put that picture into the AI tool. The and find out this man who is this idiot. Type කරලා බලන්න it will be give the output. Okay. So ask from the chat GPT. So it will be give who am I? Right. Okay. Okay. So it is very intelligent now. So it will be give all the correct details. Right. But sometimes it will make some mistakes. Okay. Right. Okay. So we will go to the next part. I will go for a chromosome genetic disorders due to the sex-linked inheritance disorders, hemophilia, color blindness, and gene mutation-related disorders, thalassemia or chromosome mutations. So applications of knowledge inheritance, those part in a separate day because now the time is the limited time here because I may rent out the places to do this online session because in my home there is a internet issue. That's why I came to here that this is a new place. So tell me about the experience about today class. So we use the whiteboard and we use the digital screens also. So tell me your experiences and what is the I need your feedbacks, what are the suggestions, what we should improve and what you expect more from me. So put into the WhatsApp group or WhatsApp community, or privately you can WhatsApp me. So you all have WhatsApp number. If you don't have a number, please write down note 7063532008. Okay, so this is my WhatsApp number. If you have any doubt, you can WhatsApp me. So in the second part of this session, I will continue in a other day. It will be soon as possible I will do. So you can WhatsApp me if you have any doubt. So I will end the sessions from now in here. So I will send some MCQ questions and some essay questions and past paper questions what are the available in your previous papers. So I will collect all them and I will put into your WhatsApp community so you can take them and practice. So this is today seminar. So from here I will stop. So if you have any doubt, you can ask me. Okay, සර්ගේ හොඳම ගෝලයා කවුද පන්තියේ? ගෝලයා ගොඩක් ඉන්නවා. නේද? හොඳම ගෝලයා ගොඩක් ඉන්නවා. මේ at the end of the classes මේ කියන්නේ අන්තිමේටම class එක ඉවර කරන දවසක් තියෙනවානේ. So when you go to the exams, so at that time I will tell you හොඳම ගෝලයා කවුද කියලා. Right? Okay. එහෙනම් thank you for your joining. මේ if මට චූටි උදව්වක් ඕන ඔයාලගෙන්. So if you take something from your class, tell me about this class to your friends as well. So most of the people don't know about me because neither I am not doing much advertising. So you are my helpers neither. So you are my students. So you give this message to your friends and help me to neither find more students. Okay, right. So thank you for your joining. And now see you, Iboan, and see you in the next class. See you.